Welcome to this quiet moment of learning, where we'll embark together on a soothing journey through one of your body's most remarkable systems. Let yourself settle comfortably as we begin to explore blood, not merely as that familiar red liquid you glimpse during a small cut, but as something far more profound and beautiful. Take a slow, deep breath and allow your mind to drift into a state of calm curiosity. We're going to discover blood as a specialized connective tissue, the only fluid tissue in your entire body. This may seem like a simple distinction, but it reveals something extraordinary about the substance flowing through you right now. While other connective tissues like bone, cartilage, and the fibrous networks that hold your organs in place provide structure and support, blood moves. It flows, it carries, it connects every distant corner of your being in a continuous living current. Picture, if you will, blood as a vast, intricate river system. Just as mighty rivers branch into smaller streams that eventually reach the most remote valleys and meadows, your blood flows from your heart through progressively smaller vessels, eventually reaching capillaries so tiny that red blood cells must travel through them single file. This liquid foundation touches every cell in your body, creating an intimate connection between your heart and the furthest reaches of your fingertips, the depths of your organs, and even the delicate tissues behind your eyes. Let's pause here for a moment and consider the sheer volume of this remarkable fluid within you. Blood constitutes approximately 8% of your total body weight, a significant portion when you truly think about it. If you're an average-sized male, you're carrying about 5 to 6 liters of blood. If you're female, it's typically 4 to 5 liters. To help you visualize this, imagine several large water bottles, their contents transformed into this complex, living fluid that sustains every moment of your existence. Don't worry if these numbers feel overwhelming right now. We'll return to them gently throughout our journey, and they'll begin to feel familiar, like old friends. What's important now is simply appreciating the presence of this substantial volume of life-sustaining liquid within you, quietly doing its work as you rest and learn. Your blood maintains a slightly alkaline nature, with a pH that hovers between 7.35 and 7.45. This might seem like a narrow range, but it's precisely calibrated for optimal function. Think of this pH balance as a delicate dance, carefully maintained by your body's wisdom. Even slight deviations from this range would disrupt the intricate chemical processes that keep you alive, so your body works constantly and quietly to maintain this perfect balance. The temperature of your blood runs just slightly warmer than your core body temperature, typically around 38 degrees Celsius or about 100.4 degrees Fahrenheit. This extra degree of warmth comes from the gentle friction created as blood flows through countless vessels, and from the metabolic heat generated by the organs it passes through. It's a subtle reminder that blood is not a passive fluid, but an active participant in your body's thermal regulation. As we prepare to delve deeper into blood's composition and functions in the sections ahead, let this foundation settle gently in your mind. Blood is simultaneously a transport medium, carrying oxygen, nutrients, hormones, and waste products where they need to go, and a communication network, enabling distant parts of your body to coordinate with one another. It's the liquid thread that weaves through every organ system, creating the unified, harmonious functioning that is your life. Allow yourself to feel a quiet sense of wonder at this fluid foundation flowing through you right now. Every heartbeat sends fresh waves of this life-sustaining current throughout your body, delivering what your cells need and carrying away what they no longer require. This process continues as you sleep, as you wake, as you learn, a constant, faithful service that connects every part of you in perfect, quiet harmony. Let this gentle understanding prepare your mind for the deeper explorations ahead as we continue our peaceful journey through the remarkable world of blood. As we continue our peaceful journey together, let your mind settle even more deeply into this state of calm learning. We're going to explore the most abundant component of your blood, plasma, the remarkable liquid medium that makes up over half of your blood's volume and serves as the foundation for all other blood functions. Picture plasma as a serene, golden ocean within your vessels, a nutrient-rich sea in which your blood cells gently float and travel on their vital missions. This isn't merely water with a few dissolved substances. Plasma is a complex, carefully balanced solution that represents one of nature's most sophisticated transport systems. As you breathe quietly now, imagine this golden liquid flowing smoothly through your arteries and veins, carrying its precious cargo to every corner of your being. Plasma consists of approximately 90% water, but within that water lies a treasure trove of dissolved substances that make life possible. Think of plasma as your body's internal postal service, constantly in motion, never resting in its dedication to maintaining the delicate balance that keeps you alive. The remaining 10% of plasma contains proteins, nutrients, electrolytes, hormones, gases, and waste products, each component present in precisely the right concentration to support optimal cellular function. Let's begin with plasma proteins, the most abundant dissolved substances in this liquid medium. These remarkable molecules serve multiple functions, and understanding them will help you appreciate the elegant complexity of the fluid flowing through you right now. The three major categories of plasma proteins work together in perfect harmony: albumin, globulins, and fibrinogen. Albumin, the most abundant plasma protein, acts as your body's molecular sponge. Imagine tiny protein molecules constantly working to maintain the proper balance of fluids between your blood vessels and the surrounding tissues. Albumin creates what we call oncotic pressure, a gentle pulling force that helps keep fluid within your blood vessels rather than allowing it to leak out into your tissues. Without adequate albumin, you would experience swelling as fluid accumulates where it shouldn't be. This protein also serves as a transport vehicle, carrying hormones, fatty acids, and certain medications through your bloodstream to their intended destinations. Globulins represent a diverse family of proteins, each with specialized functions. Some globulins serve as antibodies, your immune system's molecular guards that recognize and neutralize foreign substances. Others act as transport proteins, carefully carrying lipids, hormones, and metals through your plasma. Think of globulins as a specialized workforce, each type perfectly designed for its particular role in maintaining your health and well-being. Fibrinogen, the third major plasma protein, remains quietly dissolved in your plasma until the moment it's needed for blood clotting. When injury occurs, fibrinogen transforms into fibrin, creating the mesh-like structure that forms the foundation of blood clots. We'll explore this remarkable transformation in greater detail later in our journey, but for now, simply appreciate fibrinogen as a molecular tool that remains patiently ready to spring into action whenever your body needs repair. Beyond proteins, plasma carries the nutrients that fuel your cellular metabolism. Glucose, the primary sugar that powers your cells, maintains steady levels in plasma through careful regulation by your liver and pancreas. Amino acids, the building blocks of proteins, circulate in plasma after being absorbed from your digestive system, ready to be delivered to cells that need to build or repair proteins. Lipids travel through plasma in special packages called lipoproteins, ensuring that fat-soluble vitamins and essential fatty acids reach their destination safely. The electrolyte composition of plasma maintains the electrical properties necessary for nerve conduction and muscle contraction. Sodium, potassium, calcium, magnesium, chloride, and bicarbonate exist in precisely balanced concentrations, creating the ionic environment that allows your cells to communicate with each other. Think of these electrolytes as the electrical infrastructure of your body, enabling the flow of information that coordinates every physiological process. Dissolved gases in plasma include not only carbon dioxide being transported away from your tissues, but also small amounts of oxygen, nitrogen, and other atmospheric gases. The carbon dioxide dissolved in plasma plays a crucial role in maintaining your blood's pH balance, working as part of a sophisticated buffering system that keeps your internal environment stable. Hormones travel through plasma like molecular messengers, carrying instructions from endocrine glands to target tissues throughout your body. Insulin from your pancreas, thyroid hormones from your neck, cortisol from your adrenal glands, all of these chemical signals dissolve into plasma and travel to cells that have specific receptors waiting to receive their messages. Finally, plasma carries waste products away from your cells toward organs capable of elimination. Urea, created when proteins are broken down, travels through plasma to your kidneys for removal. Bilirubin, produced when old red blood cells are recycled, journeys through plasma to your liver for processing. This constant cleaning service ensures that cellular waste products never accumulate to harmful levels. As we prepare to explore the cellular components of blood in our next section, take a moment to appreciate plasma's quiet elegance. This golden liquid medium creates the perfect environment for cellular life, maintaining optimal conditions for the countless chemical reactions that sustain you. Every heartbeat circulates this remarkable fluid, carrying life-sustaining substances to your cells while simultaneously removing what they no longer need. Allow yourself to feel grateful for this liquid foundation that makes all other blood functions possible. The calm, nutrient-rich ocean that has been faithfully serving you every moment of your life, creating the perfect medium for the cellular passengers we'll meet next in our continuing journey. Let this understanding of plasma's vital role settle gently in your mind as we prepare to explore the remarkable cells that travel within this golden sea. As we continue our peaceful journey together, let your breathing slow and deepen as we turn our attention to the most abundant residents of your bloodstream, the remarkable red blood cells or erythrocytes. These tiny cellular messengers make up nearly half of your blood's volume, and as you rest comfortably now, billions of them are quietly flowing through your vessels, carrying out their singular vital mission. Picture these cells as microscopic flexible discs, shaped remarkably like tiny donuts without holes, what scientists call a biconcave shape. This elegant design isn't accidental, it's the result of millions of years of evolutionary refinement. Allow your mind to visualize these cells as they flow through your circulatory system right now, constantly moving, never resting, each one perfectly designed for its life-sustaining purpose. The biconcave shape of red blood cells creates several important advantages. First, this dimpled design maximizes the cell's surface area relative to its volume, providing more space for gas exchange to occur. Think of it like increasing the number of windows in a house. More surface area means more opportunities for oxygen to enter and carbon dioxide to leave. Second, this flexible shape allows red blood cells to squeeze through the narrowest capillaries in your body, some so small that these cells must fold and deform to pass through single file. What makes these cells even more remarkable is what they've given up to serve you better. Unlike almost every other cell in your body, mature red blood cells lack a nucleus. They've essentially sacrificed their cellular headquarters to make more room for their precious cargo. They've also eliminated most of their internal organelles, those tiny cellular machines that normally maintain cell function. This represents a beautiful example of cellular specialization and sacrifice. Red blood cells have streamlined themselves to excel at one crucial task: carrying oxygen. These dedicated cells live for approximately 120 days, during which time they make countless journeys throughout your body. Imagine each red blood cell as a tireless courier, making perhaps 1,000 round trips from your lungs to distant tissues and back again during its 4-month lifespan. They travel through your largest arteries and your tiniest capillaries, never complaining, never resting, constantly working to keep every cell in your body supplied with the oxygen it needs to function. The magic that enables red blood cells to carry oxygen lies in a remarkable protein called hemoglobin. Each red blood cell contains approximately 280 million hemoglobin molecules, and each hemoglobin molecule can carry up to 4 oxygen atoms. This means that a single red blood cell can transport over 1 billion oxygen atoms, a truly staggering number when you consider that you have approximately 25 trillion red blood cells circulating through your body right now. Hemoglobin gives blood its distinctive red color, but this color actually changes depending on whether the hemoglobin is carrying oxygen or not. When hemoglobin binds with oxygen in your lungs, it creates oxyhemoglobin, which has a bright, vibrant red color. This is why arterial blood that has just picked up oxygen from your lungs, appears so brilliantly red, When hemoglobin releases its oxygen to hungry tissues, it becomes deoxyhemoglobin, which has a darker, more purplish red color. This is why venous blood returning to your heart after delivering oxygen, appears darker. The structure of hemoglobin itself is a marvel of molecular engineering. Each hemoglobin molecule contains 4 protein chains wrapped around 4 ion-containing groups called hemogroups. It's the iron atoms in these hemogroups that actually bind to oxygen molecules. Think of iron as nature's chosen element for this crucial task. Its unique chemical properties make it perfect for grabbing onto oxygen when it's abundant in your lungs and releasing it when it's needed in your tissues. Red blood cells are produced in your bone marrow through a process called erythropoiesis, which we'll explore more deeply later in our journey. What's important to understand now is that your body is constantly producing new red blood cells to replace those that have completed their 120-day service. Every second, your bone marrow produces approximately 2.5 million new red blood cells, a production rate that would be the envy of any manufacturing facility. The concentration of red blood cells in your blood is measured as a hematocrit, the percentage of your blood volume that consists of red blood cells. For men, this typically ranges from 41% to 50%, while for women, it's usually between 36% and 44%. This means that nearly half of your blood volume consists of these oxygen-carrying specialists, highlighting just how important their function is to your survival. Red blood cells also play a role in maintaining your blood's pH balance. They contain an enzyme called carbonic anhydrase, which helps convert carbon dioxide, a waste product of cellular metabolism, into bicarbonate ions that can be transported in your plasma. This process not only helps remove waste from your tissues, but also helps maintain the slightly alkaline pH of your blood that we discussed in our first section. As we prepare to follow these remarkable cells on their oxygen-carrying journey in our next section, take a moment to appreciate the elegant simplicity of their design. Red blood cells represent one of nature's most successful cellular specialists, cells that have given up complexity to achieve mastery of a single crucial function. They flow through you right now with quiet dedication, each one a microscopic servant of life, ensuring that every breath you take translates into oxygen delivered where it's needed most. Allow yourself to feel grateful for these billions of cellular couriers working tirelessly within you. Their flexible forms flowing through vessels both wide and narrow, their iron-rich cores carrying the breath of life to every corner of your being. In the next section of our gentle journey, we'll follow them on their remarkable voyage from lung to cell, understanding the beautiful process that connects your breathing with the cellular respiration happening throughout your body. Let this appreciation for these remarkable oxygen carriers settle peacefully in your mind as we prepare to witness their vital journey through your circulatory system. Let yourself sink deeper into this state of calm learning as we follow oxygen on one of nature's most essential journeys. Right now, as you breathe quietly, millions of oxygen molecules are beginning an extraordinary voyage that will take them from the air around you to the most distant cells in your body. This journey represents one of life's most fundamental processes, connecting each breath you take with the cellular respiration happening in every corner of your being. Imagine each breath you take as the beginning of countless tiny deliveries, with hemoglobin molecules acting as gentle molecular couriers. When air enters your lungs, it travels through progressively smaller passages until it reaches the alveoli, microscopic air sacs, where the magic of gas exchange occurs. Picture these alveoli as tiny delicate balloons, their thin walls creating the perfect interface between the air you've breathed and the blood flowing through your pulmonary capillaries. The moment oxygen crosses from your lungs into your bloodstream, it encounters red blood cells loaded with hemoglobin molecules, eagerly waiting to receive their precious cargo. This binding process is remarkably gentle and reversible. Oxygen doesn't become permanently attached to hemoglobin, but rather forms a loose, temporary association that can be easily released when and where it's needed most. When oxygen binds to the iron atoms within hemoglobin, something beautiful happens. The iron changes its electronic structure slightly, and this change is what creates the brilliant red color of arterial blood. Think of this color change as hemoglobin's way of announcing that it's fully loaded and ready for its delivery mission. Each hemoglobin molecule can carry up to 4 oxygen atoms, and when fully saturated, your arterial blood becomes that vibrant, life-affirming red that symbolizes vitality and health. As oxygen-rich blood leaves your lungs, it begins its journey through your arterial system. Your heart serves as the central pumping station, sending this freshly oxygenated blood through your aorta, your body's main highway, and then into progressively smaller arteries that branch like a vast tree throughout your body. The pressure created by your heart's contraction propels this oxygen-rich blood toward tissues that are constantly consuming oxygen for their metabolic processes. The remarkable thing about hemoglobin is how intelligently it releases its oxygen cargo. This release isn't random, it's precisely controlled by the local conditions in your tissues. In areas where cells are working hard and consuming lots of oxygen, the environment becomes slightly more acidic, the temperature may rise slightly, and carbon dioxide levels increase. These changes signal to hemoglobin that oxygen is needed here, and it responds by releasing its precious cargo more readily. This process, known as the Bohr effect, represents one of nature's most elegant feedback systems. Areas of high metabolic activity, like working muscles or busy organs, create the exact conditions that encourage hemoglobin to release more oxygen. It's as if hemoglobin can sense where oxygen is needed most and adjusts its grip accordingly. Tissues at rest, which don't need as much oxygen, don't create these signals, so hemoglobin holds onto its oxygen more tightly as it passes through. As hemoglobin releases its oxygen, its color gradually changes from bright red to a darker, more purplish hue. This color change reflects the molecular transformation happening within the iron atoms. They're returning to their original state as oxygen is released. This is why venous blood, which has delivered much of its oxygen to hungry tissues, appears darker than arterial blood. Don't worry if the chemistry feels complex. What's important is understanding this as a natural, ongoing dance that happens billions of times each day without any conscious effort on your part. The oxygen that hemoglobin releases doesn't simply float around in your tissues, it dissolves into the fluid surrounding your cells and then diffuses across cell membranes, where it becomes available for cellular respiration. Inside your cells, oxygen participates in the intricate process of energy production, helping to convert glucose and other nutrients into the ATP that powers cellular activities. This is where the oxygen from your breath literally becomes the energy that sustains your life. Meanwhile, as cells consume oxygen, they produce carbon dioxide as a waste product. Some of this carbon dioxide dissolves directly into your blood plasma, but a significant portion actually binds to hemoglobin as well, though to a different part of the molecule than oxygen. This means that hemoglobin serves a dual purpose, delivering oxygen to tissues and helping to carry carbon dioxide away from them. The carbon dioxide that binds to hemoglobin doesn't interfere with oxygen binding. The two gases use different attachment sites on the hemoglobin molecule. However, when carbon dioxide levels are high, it does encourage hemoglobin to release oxygen more readily, which is exactly what you want in metabolically active tissues that are producing lots of carbon dioxide waste. As deoxygenated blood returns to your heart through your venous system, it carries this carbon dioxide back to your lungs, where the process reverses. In your lungs, where oxygen levels are high and carbon dioxide levels are lower, hemoglobin readily releases carbon dioxide and picks up fresh oxygen, beginning the cycle anew. The carbon dioxide is then exhaled, completing this beautiful cycle of gas exchange. This entire process represents one of life's most fundamental exchanges, connecting your breathing with the cellular respiration happening throughout your body. Every breath you take initiates millions of these molecular deliveries, ensuring that your cells receive the oxygen they need while simultaneously removing the carbon dioxide they no longer require. As we prepare to explore the protective forces that patrol your bloodstream in our next section, allow yourself to appreciate this remarkable process that's happening within you right now. With each gentle breath you take, you're initiating countless molecular journeys, each one essential to maintaining the cellular activities that sustain your life. This process continues whether you're awake or asleep, active or at rest, a constant, faithful exchange that connects the air around you with the most intimate processes of cellular life. Let this understanding of oxygen's vital journey settle peacefully in your mind as we prepare to meet the guardian cells that protect this precious transport system. As we continue our peaceful journey through the world of blood, allow your breathing to remain slow and steady as we meet the protective forces that patrol your bloodstream, the white blood cells, or leukocytes. These remarkable cells represent your body's sophisticated security system, a quiet but vigilant force that works tirelessly to maintain your health while you rest, learn, and live your daily life. Unlike the abundant red blood cells we've just explored, white blood cells are far fewer in number. You have only about one white blood cell for every 600 to 700 red blood cells. Yet despite their relative scarcity, these cells wield enormous influence over your health and survival. Think of them as an elite security force rather than a large army, small in number but highly trained and incredibly effective at their specialized roles. White blood cells differ fundamentally from red blood cells in several important ways. While red blood cells gave up their nuclei to make room for oxygen-carrying hemoglobin, white blood cells retain their cellular command centers, allowing them to respond to changing situations, communicate with other cells, and even reproduce when needed. They're also capable of leaving your bloodstream entirely, squeezing between the cells that line your blood vessels to patrol the tissues beyond. Let us gently meet the different types of white blood cells, each with its own specialized role in protecting you. The most abundant are the neutrophils, making up about 50 to 70% of all white blood cells. Picture these cells as your body's first responders, the cellular equivalent of emergency medical technicians who arrive quickly at sites of infection or injury. Neutrophils live relatively short lives, only about 6 to 8 hours in your bloodstream, but they're constantly being replenished from your bone marrow. When bacteria invade your tissues, or when you suffer an injury, neutrophils are among the first immune cells to arrive at the scene. They can engulf and destroy bacteria through a process called phagocytosis, literally meaning cell eating. Imagine these cells as microscopic cleanup crews, patrolling your body and removing harmful invaders and cellular debris. When neutrophils encounter something that shouldn't be there, they sacrifice themselves in the process of neutralizing the threat, forming what we commonly recognize as pus. Next, we meet the lymphocytes, which make up about 20 to 30% of your white blood cells. These cells represent your body's intelligence network and specialized forces. There are several types of lymphocytes, but the two most important are B cells and T cells. B cells function like biological weapons manufacturers, producing antibodies, specialized proteins designed to recognize and neutralize specific threats. When a B cell encounters a foreign substance, it can transform into a plasma cell that produces thousands of identical antibodies, each precisely designed to target that particular invader. T cells serve multiple roles in your immune system. Some T cells, called helper T cells, coordinate immune responses by communicating with other immune cells and helping to activate them. Think of helper T cells as battlefield commanders, assessing threats and directing other immune forces to respond appropriately. Other T cells, called cytotoxic T cells, directly attack cells that have been infected by viruses or have become cancerous. These cellular assassins can recognize when your own cells have been compromised and eliminate them before they can cause further harm. What makes lymphocytes particularly remarkable is their memory. Once they've encountered a specific threat, some lymphocytes transform into memory cells that remain in your system for years, sometimes for your entire life. These memory cells allow your immune system to respond much more quickly and effectively if the same threat appears again. This is the principle behind vaccination and why you typically only get diseases like chickenpox once. Monocytes, making up about 4 to 8% of white blood cells, are the largest of the white blood cells circulating in your bloodstream. Think of monocytes as traveling supervisors who patrol your circulatory system but can transform when needed. When monocytes leave your bloodstream and enter tissues, they mature into macrophages, literally meaning big eaters. These large cells serve as both cleanup crews and antigen processors, engulfing cellular debris, dead cells, and foreign particles, while also breaking down complex substances into smaller pieces that other immune cells can recognize and respond to. Macrophages also play crucial roles in tissue repair and maintenance. They help remove damaged cells after injury, clear away old red blood cells that have completed their 120-day lifespan, and even assist in the remodeling of tissues during healing. Picture them as gentle but thorough janitors, constantly working to keep your tissues clean and functional. The remaining white blood cells, eosinophils and basophils, make up only small percentages of your total white blood cell count, but they have specialized roles that become crucial under certain circumstances. Eosinophils are particularly important in defending against parasitic infections and are often elevated in people with allergies or asthma. Basophils release histamine and other inflammatory mediators, playing important roles in allergic reactions and the inflammatory response. Unlike red blood cells, which live for about 120 days, white blood cells have varying lifespans depending on their type and function. Some neutrophils live only hours, while certain memory lymphocytes can survive for decades. This variation reflects their different roles. Short-lived cells like neutrophils are designed for immediate response and are constantly being replenished, while long-lived memory cells provide lasting protection based on past encounters with threats. White blood cells also differ from red blood cells in their movement patterns. While red blood cells simply flow with the current of your bloodstream, white blood cells can move independently, changing shape and squeezing through tissue spaces to reach areas where they're needed. They can follow chemical signals that lead them to sites of infection or injury, a process called chemotaxis, chemical attraction that guides these cellular guardians exactly where they need to go. As we prepare to explore the smallest cellular components of blood in our next section, take a moment to appreciate these microscopic protectors working within you right now. They patrol your bloodstream and tissues with quiet diligence, ready to respond to threats you may never even notice. Some are destroying harmful bacteria, others are producing antibodies against potential future threats, and still others are cleaning up cellular debris and maintaining the health of your tissues. Allow yourself to feel grateful for this sophisticated security system that requires no conscious direction from you. These cellular guardians work tirelessly while you sleep, learn, and go about your daily activities, representing one of nature's most elegant solutions to the challenge of maintaining health in a world full of potential threats. Their presence ensures that the oxygen-carrying red blood cells and nutrient-rich plasma we've explored can continue their vital work in a safe, protected environment. Let this appreciation for your body's silent guardians settle peacefully in your mind as we prepare to meet the smallest but equally important cellular components of your blood. As we continue our gentle journey through the remarkable world of blood, let yourself settle even more deeply into this state of calm learning. We now turn our attention to the smallest formed elements in your blood, platelets or thrombocytes. These tiny cellular fragments may be small in size, but they play an enormous role in keeping your circulatory system intact and functional. Picture platelets as microscopic repair specialists, constantly circulating through your bloodstream like a dedicated maintenance crew. Unlike the red blood cells and white blood cells we've explored, platelets aren't actually complete cells, they're small fragments of much larger cells, each one carrying within it the molecular tools necessary for one of your body's most important functions, stopping bleeding when blood vessels are damaged. These remarkable structures are actually fragments of giant cells called megakaryocytes that reside in your bone marrow. Imagine these megakaryocytes as cellular factories, each one capable of producing thousands of platelets before fragmenting and releasing them into your circulation. A single megakaryocyte can generate anywhere from 1,000 to 3,000 platelets, representing one of nature's most efficient production systems. Once released into your bloodstream, platelets have a relatively short lifespan of about 8 to 10 days. During this time, they circulate quietly through your vessels, maintaining a state of readiness that allows them to spring into action the moment they're needed. Think of them as molecular first aid kits, each one containing the essential components needed to begin the healing process when injury occurs. Under normal circumstances, platelets remain inactive as they flow through your bloodstream. They're smooth and disc-shaped, similar to red blood cells but much smaller, so small that you could fit about 20 platelets across the diameter of a single red blood cell. This small size allows them to flow easily through even the tiniest capillaries while remaining ready to respond instantly to vascular damage. The moment platelets encounter damaged blood vessel walls, they undergo a remarkable transformation. When the smooth inner lining of a blood vessel is torn or cut, it exposes underlying tissues that platelets recognize as abnormal. This exposure triggers platelets to change from their inactive, smooth form into an active, sticky configuration with long, thin projections called pseudopodia, literally meaning false feet, that help them adhere to the damaged area and to each other. This activation process represents one of your body's most rapid emergency responses. Within seconds of encountering damage, platelets begin to stick to exposed collagen fibers in the blood vessel wall, forming what's called a platelet plug. Think of this as applying a living bandage that forms automatically wherever it's needed. Approximately 150,000 to 400,000 platelets circulate in each microliter of your blood. That's 150 to 400 million in just a single drop. This enormous number ensures that wherever damage occurs in your vast circulatory network, sufficient platelets are immediately available to begin the repair process. It's a vast army of microscopic repair workers distributed throughout your entire circulatory system and ready to respond within moments to any breach in your vascular walls. Platelets contain several types of granules filled with different substances that aid in the clotting process. Dense granules contain molecules like adenosine diphosphate, ADP, and serotonin that help attract more platelets to the site of injury and enhance their stickiness. Alpha granules contain proteins and growth factors that not only assist in clot formation, but also promote healing and tissue repair once the immediate crisis has passed. When platelets become activated, they release the contents of these granules in a process called degranulation. This release serves multiple purposes. It attracts additional platelets to the area, enhances the stickiness of nearby platelets, and begins the complex cascade of reactions that will ultimately form a stable blood clot. Think of this as platelets sending out molecular distress signals that recruit reinforcements and prepare the area for more extensive repair. The surface of activated platelets also becomes studded with receptor proteins that can bind to various clotting factors, essentially transforming each platelet into a tiny platform where the more complex reactions of blood coagulation can occur. We'll explore this intricate process in our next section, but for now, simply appreciate how platelets serve as both the initial response to vascular injury and the foundation upon which more sophisticated repair mechanisms can build. Platelets also play roles beyond hemostasis, the process of stopping bleeding. They release growth factors that promote the repair and regeneration of damaged tissues, and they interact with white blood cells in ways that can influence immune responses and inflammation. Some research even suggests that platelets may play roles in maintaining the health and integrity of blood vessel walls under normal circumstances. The production and regulation of platelets is carefully controlled by your body through hormones like thrombopoietin, which stimulates megakaryocyte production in your bone marrow when platelet levels drop. This ensures that you maintain adequate numbers of these cellular repair specialists at all times, ready to respond to both minor everyday injuries and more significant vascular damage. As we prepare to explore the orchestrated process of hemostasis in our next section, take a moment to appreciate these tiny fragments working within you right now. They flow silently through your circulation, smaller than your other blood cells, yet carrying within them the essential tools for maintaining the integrity of your entire circulatory system. Every small cut that stops bleeding, Every minor injury that heals without significant blood loss represents the successful work of these microscopic healers. Allow yourself to feel grateful for these dedicated cellular fragments that require no conscious direction from you, yet stand ready to protect you from blood loss every moment of every day. Their presence ensures that the complex transport systems we've explored, the oxygen-carrying red blood cells, the protective white blood cells, and the life-sustaining plasma, can continue their vital work within the secure environment of intact blood vessels. Let this appreciation for these gentle healers settle peacefully in your mind as we prepare to witness the remarkable orchestrated response that occurs when your vascular system needs repair. As we continue our peaceful journey through the remarkable world of blood, let yourself breathe even more slowly and deeply as we witness one of your body's most beautifully orchestrated emergency responses. We're going to explore hemostasis, the elegant three-step process that stops bleeding when blood vessels are injured. Think of this as a perfectly choreographed molecular symphony that begins playing the moment any part of your circulatory system experiences damage. This process happens so seamlessly and automatically that you rarely notice the constant maintenance work occurring within your body. Every small cut that stops bleeding, every minor bump that doesn't result in significant blood loss, these represent the successful coordination of multiple biological systems working together in perfect harmony. Allow your mind to settle into appreciating this remarkable process that protects the precious fluid we've been exploring together. The three phases of hemostasis unfold like movements in a classical symphony, each building upon the previous one while maintaining the overall theme of protection and repair. These phases are vascular spasm, platelet plug formation, and coagulation, each one essential, each one precisely timed, and each one contributing to the successful preservation of your circulatory integrity. The first movement, vascular spasm. When a blood vessel is injured, the very first response occurs within seconds before platelets can even reach the scene, before any clotting factors can be activated. The smooth muscle cells that surround your blood vessels immediately contract, creating what we call a vascular spasm. Picture this as your blood vessel's immediate defensive reaction, like reflexively pulling your hand away from something hot. This contraction serves several important purposes. First, it directly reduces the diameter of the damaged vessel, immediately decreasing blood flow through the injured area. Think of this as pinching a garden hose to reduce the water flow. The same principle applies to your blood vessels. This vasoconstriction can be so effective that very small cuts may stop bleeding entirely during this phase, buying precious time for the more sophisticated repair mechanisms to begin their work. The vascular spasm also brings the edges of damaged tissue closer together, making it easier for platelets to form an effective seal and for the later stages of clotting to create a stable repair. This response can last anywhere from several minutes to several hours, depending on the extent of the injury and the size of the blood vessel involved. What triggers this immediate response? The injury itself causes the release of chemicals from damaged tissues, and the smooth muscle cells in blood vessel walls respond to these signals by contracting. Additionally, platelets that begin arriving at the scene release substances like serotonin and thromboxane A2, which further enhance and maintain the vascular spasm. It's as if your body has multiple backup systems, ensuring that this first line of defense remains active long enough for reinforcements to arrive. The second movement, platelet plug formation. While the vascular spasm provides immediate damage control, the second phase of hemostasis involves the platelets we met in our previous section. These tiny cellular fragments quickly transform from passive passengers in your bloodstream into active participants in vascular repair. When platelets encounter the exposed collagen fibers and other substances normally hidden beneath the smooth inner lining of blood vessels, they undergo the remarkable transformation we discussed earlier. The process begins with platelet adhesion, individual platelets sticking to the damaged vessel wall. This adhesion is mediated by a protein called von Willebrand factor, which acts like molecular glue, helping platelets attach firmly to exposed collagen. Think of von Willebrand factor as creating tiny molecular bridges that allow platelets to gain a firm foothold on the damaged vessel wall, even in areas where blood flow might otherwise wash them away. Once platelets adhere to the injury site, they become activated and begin to change shape, extending those thin projections called pseudopodia that we mentioned earlier. These projections help platelets spread across the damaged area and begin to stick to each other in a process called platelet aggregation. As more platelets arrive and become activated, they release chemical signals that attract even more platelets, creating a positive feedback loop that rapidly builds a platelet plug at the site of injury. The activated platelets also release adenosine diphosphate, ADP, from their dense granules, which makes nearby platelets more sticky and more likely to aggregate. They also release thromboxane 2, which not only enhances platelet aggregation but also helps maintain the vascular spasm we discussed in the first phase. This represents beautiful biological coordination, each phase of hemostasis supporting and enhancing the others. Within minutes, this growing mass of activated platelets can effectively seal small breaks in blood vessels. For tiny injuries like those caused by normal wear and tear on your capillaries, this platelet plug may be sufficient to stop bleeding entirely. However, for larger injuries, this plug serves as the foundation for the third and most complex phase of hemostasis. The third movement, the coagulation cascade. The final phase of hemostasis involves the transformation of the temporary platelet plug into a stable, permanent clot through the process of coagulation. This represents one of biology's most intricate molecular cascades, involving more than a dozen different proteins called clotting factors that work together like a precisely choreographed assembly line. Think of coagulation as your body's way of reinforcing the temporary repair created by the platelet plug. While the platelet plug can stop bleeding quickly, it's relatively fragile and temporary. The coagulation cascade creates a mesh of strong protein fibers that traps blood cells and creates a stable, long-lasting seal that can withstand the normal pressures and movements of daily life. The coagulation cascade can be initiated through two pathways, the intrinsic pathway and the extrinsic pathway. But both pathways converge on a common final sequence that ultimately converts the soluble plasma protein fibrinogen into insoluble fibrin fibers. Don't worry about memorizing these pathway names. What's important is understanding that your body has multiple ways to trigger this life-saving process, ensuring that clotting can begin even if one pathway is compromised. The extrinsic pathway is triggered when tissue factor, a protein normally hidden inside cells, is exposed to blood due to tissue damage. This pathway provides a rapid response to injury and is the primary mechanism for initiating clotting in most bleeding situations. The intrinsic pathway can be activated by contact with foreign surfaces or damaged blood vessel walls and serves as a backup system that can maintain clotting even when tissue factor levels are low. Both pathways involve a series of enzymatic reactions where each clotting factor, when activated, serves as an enzyme that activates the next factor in the sequence. This creates what biochemists call a cascade, a series of amplifying reactions where each step produces more active enzymes than were present in the previous step. The result is that a small initial signal can rapidly generate a large response, ensuring that clot formation occurs quickly enough to prevent dangerous blood loss. The key players in this final common pathway include prothrombin, which is converted to thrombin, and fibrinogen, which thrombin converts to fibrin. Thrombin serves as both an enzyme that creates fibrin fibers and a powerful activator of platelets and several clotting factors, creating multiple positive feedback loops that ensure rapid and effective clot formation once the process begins. Fibrin fibers form a mesh-like network that traps red blood cells, white blood cells, and platelets, creating the familiar red clot that we recognize as the final product of successful hemostasis. This clot is initially soft and somewhat fragile, but over time it undergoes a process called clot retraction, where platelets within the clot contract and pull the fibrin fibers tighter, creating a more compact and stable seal. As we prepare to explore the birthplace where all these remarkable blood cells begin their lives, Take a moment to appreciate this orchestrated emergency response that's available to you every moment of every day. The three phases of hemostasis represent one of your body's most successful collaborations between different cellular and molecular systems, each contributing their specialized capabilities to maintain the integrity of your circulatory system. Allow yourself to feel grateful for this automatic protection that requires no conscious thought or direction from you. Whether you're aware of it or not, minor injuries to your blood vessels occur regularly as part of normal daily activities, and this elegant three-step process quietly repairs them, ensuring that your precious blood remains safely contained within its designated pathways, while continuing its vital work of sustaining every cell in your body. Let this appreciation for your body's remarkable repair mechanisms settle peacefully in your mind as we prepare to explore the hidden factories where all blood cells begin their extraordinary lives. As we continue our peaceful exploration through the remarkable world of blood, let yourself settle even more deeply into this state of calm learning. We now journey to one of your body's most remarkable and hidden places, the red bone marrow, the quiet factory where every single blood cell we've encountered began its life. This process of blood cell formation, called hematopoiesis, represents one of nature's most extraordinary examples of cellular renewal and regeneration. Picture the bone marrow as a bustling but orderly city hidden within the protective walls of your bones. Unlike the organs you can feel and see, bone marrow works in complete darkness, deep within the hollow spaces of your larger bones, carrying out one of your body's most essential manufacturing processes. Right now, as you rest and learn, millions of stem cells within your bone marrow are quietly dividing, differentiating, and maturing into the various blood cells that will soon join the circulation we've been exploring together. The story of hematopoiesis begins with remarkable cells called pluripotent hematopoietic stem cells. Think of these as the ultimate cellular ancestors, each one possessing the incredible ability to give rise to any type of blood cell your body needs. These stem cells represent one of biology's most elegant solutions to the challenge of maintaining a complex multi-component system rather than having separate factories for each type of blood cell. Your body maintains a single population of master cells that can become whatever is needed most at any given time. During your earliest development, blood cell production occurred in your yolk sac, then moved to your liver and spleen as you grew in the womb. But by the time you were born, this vital function had largely relocated to your bone marrow, where it continues throughout your entire life. During childhood, nearly every bone in your body contributed to blood cell production, creating the abundant supply of blood cells needed for rapid growth and development. As you matured into adulthood, this blood-forming activity became more selective, concentrating primarily in the larger bones of your body. Your vertebrae, sternum, ribs, pelvis, and the upper portions of your arm and leg bones now serve as the primary sites of hematopoiesis. These bones contain red bone marrow, which maintains its blood-forming capability throughout life, while many of your smaller bones have transitioned to yellow bone marrow, which consists primarily of fat cells and serves more as an energy reserve. The microenvironment within red bone marrow creates the perfect conditions for blood cell development. Specialized support cells create what scientists call hematopoietic niches, tiny neighborhoods within the bone marrow where stem cells can either remain dormant, waiting for future needs, or begin the process of differentiation into mature blood cells. These niches provide not only physical support, but also the chemical signals that guide stem cell behavior, ensuring that your body produces exactly the right types and numbers of blood cells to meet current demands. The process of hematopoiesis follows orderly pathways that branch like a family tree from those original pluripotent stem cells. The first major branch point creates two distinct lineages, myeloid stem cells and lymphoid stem cells. Don't worry about memorizing these terms. Instead, allow yourself to appreciate this as nature's way of organizing the complex task of producing multiple different cell types from a single source. The myeloid lineage gives rise to red blood cells, platelets, and most types of white blood cells including neutrophils, eosinophils, basophils, and monocytes. This pathway represents the source of the oxygen-carrying specialists, the gentle healers and many of the protective guardians we've already met in our journey. Each step in this differentiation process involves stem cells becoming progressively more specialized, gradually losing their ability to become other cell types while gaining the specific characteristics they'll need for their eventual functions. Erythropoiesis, the production of red blood cells, begins when myeloid stem cells receive signals indicating that oxygen-carrying capacity needs to be increased. A hormone called erythropoietin, produced primarily by your kidneys, serves as the primary signal for red blood cell production. When your kidneys detect that oxygen levels in your blood are lower than optimal, they release more erythropoietin, which travels through your bloodstream to your bone marrow and stimulates increased red blood cell production. The journey from stem cell to mature red blood cell takes approximately 7 days and involves several distinct stages. Early in this process, the developing cells retain their nuclei and other cellular organelles, actively producing the hemoglobin that will soon become their primary cargo. As development progresses, these cells gradually lose their nuclei and most of their internal structures, transforming into the streamlined oxygen carriers we explored earlier. The final step involves the cells squeezing through tiny pores in bone marrow blood vessels, entering the circulation as reticulocytes, young red blood cells that will complete their maturation during their first day or two in the bloodstream. The production of white blood cells follows similarly orderly pathways, but with more complexity due to the variety of different white blood cell types needed. Granulopolysis produces neutrophils, eosinophils, and basophils, the white blood cells that contain specialized granules filled with different substances for different protective functions. Monocytopolysis creates the monocytes that will later mature into macrophages in your tissues. The lymphoid lineage follows a somewhat different pattern, giving rise to the various types of lymphocytes we met earlier: B cells, T cells, and natural killer cells. Interestingly, while B cells complete their development within the bone marrow, T cells migrate to your thymus gland to complete their maturation. This represents one of the beautiful examples of cooperation between different organs in creating your immune system's capabilities. Thrombopolysis, the production of platelets, involves perhaps the most unusual process in hematopoiesis. Rather than producing individual cells that then mature, this pathway creates enormous cells called megakaryocytes, some of the largest cells in your bone marrow. These giant cells then fragment, releasing thousands of platelet fragments into your circulation. A single megakaryocyte can produce 1,000 to 3,000 platelets, representing a remarkably efficient manufacturing process. The regulation of hematopoiesis involves a complex network of growth factors, hormones, and cellular signals that ensure your body produces exactly what it needs when it needs it. If you develop an infection, signals from your immune system stimulate increased production of neutrophils and other infection-fighting cells. If you experience blood loss, erythropoietin levels rise to increase red blood cell production. If your platelet count drops, thrombopoietin stimulates increased megakaryocyte production. This regulatory system also includes negative feedback mechanisms that prevent overproduction of blood cells. When sufficient numbers of any particular cell type are present in circulation, they send signals back to the bone marrow to slow production, maintaining the delicate balance that keeps your blood composition optimal for current needs. The bone marrow environment also serves as a reservoir for stem cells, maintaining populations of dormant stem cells that can be activated during times of extreme need. This reserve capacity ensures that even under conditions of severe stress or injury, your body retains the ability to regenerate its blood supply. Some stem cells divide very rarely under normal circumstances, preserving their regenerative potential for future use throughout your entire lifespan. As we prepare to explore the physical properties that make blood such an effective transport medium, take a moment to appreciate this hidden manufacturing system working within you right now. Your bone marrow produces approximately 200 billion red blood cells every day, along with billions of white blood cells and platelets. This represents one of your body's most impressive production capabilities, carried out quietly and efficiently in the protected environment within your bones. Allow yourself to feel grateful for this continuous renewal that requires no conscious thought or effort from you. Every blood cell currently flowing through your vessels began its life in these hidden factories, following the same orderly developmental pathways that have sustained life for millions of years. This process continues whether you're awake or asleep, active or at rest, ensuring that your circulatory system maintains its remarkable capabilities throughout your entire lifetime. The concept that your blood is constantly being renewed, that the cells flowing through you today are different from those that flowed through you last month, adds another layer of wonder to this system we've been exploring together. Your bone marrow represents not just a manufacturing facility, but a source of constant regeneration and renewal, ensuring that the precious fluid sustaining your life remains fresh, functional, and perfectly adapted to your current needs. Let this appreciation for your body's remarkable regenerative capabilities settle peacefully in your mind as we prepare to explore the physical properties that make blood such an effective medium for sustaining life. As we continue our peaceful journey through the remarkable world of blood, let yourself sink even deeper into this state of calm understanding. We now turn our attention to the physical properties that make blood such an extraordinarily effective transport medium, its viscosity and flow characteristics. These properties, while perhaps less dramatic than the cellular processes we've explored, are fundamental to every aspect of blood's function within your body. Think of blood viscosity as the thickness or resistance to flow that blood exhibits as it moves through your vessels. Unlike water, which flows freely and easily, blood has a consistency more similar to a light syrup, thick enough to carry its cellular passengers effectively, yet fluid enough to reach every corner of your circulatory system. This careful balance represents millions of years of evolutionary refinement, creating the perfect medium for sustaining complex life. Blood is approximately 5 times more viscous than water, and this increased thickness comes primarily from the cells and proteins suspended within it. Picture the difference between pouring water from a pitcher and pouring honey. Blood's viscosity falls somewhere between these two extremes, creating the ideal consistency for efficient circulation while maintaining the ability to transport gases, nutrients, and cellular components throughout your body. The primary contributors to blood viscosity are your red blood cells, which we explored earlier in our journey. These flexible biconcave discs must flow through vessels of all sizes, from your large aorta to capillaries so narrow that red blood cells must deform to pass through single file. The concentration of red blood cells in your blood, called hematocrit, directly influences blood viscosity. When hematocrit increases, blood becomes thicker and more resistant to flow. When it decreases, blood becomes thinner and flows more easily. Under normal circumstances, your hematocrit remains within a carefully regulated range, typically 41 to 50% for men and 36 to 44% for women. This range represents the optimal balance between oxygen carrying capacity and flow characteristics. If hematocrit becomes too high, blood becomes so thick that your heart must work much harder to pump it through your vessels. If it becomes too low, while blood flows more easily, its reduced oxygen carrying capacity means tissues may not receive adequate oxygen despite good circulation. Plasma proteins also contribute significantly to blood viscosity. The albin, globulins, and fibrinogen we discussed earlier don't just perform their specific transport and protective functions, they also influence how easily blood flows through your vessels. Higher protein concentrations increase viscosity, while lower concentrations decrease it. This is why conditions that affect protein levels can have subtle but important effects on circulation. Temperature plays a fascinating role in blood viscosity, and understanding this helps explain some of your body's responses to different environmental conditions. As temperature increases, blood viscosity decreases, blood becomes thinner and flows more easily when warm. Conversely, as temperature drops, blood becomes more viscous and resistant to flow. This is why your extremities, fingers, toes, nose can feel cold and may appear pale in cold weather. The increased viscosity of cooler blood makes it more difficult to maintain robust circulation to these distant areas. Your body maintains blood temperature at approximately 38 degrees Celsius, about 100.4 degrees Fahrenheit, which is roughly 1 degree warmer than your core body temperature. This slight temperature elevation results partly from the friction generated as blood flows through your vessels, and partly from the metabolic heat produced by organs that blood passes through, particularly your liver and muscles during activity. This elevated temperature helps maintain optimal blood viscosity for efficient circulation. Dehydration has a profound impact on blood viscosity, demonstrating the crucial importance of adequate fluid intake for cardiovascular health. When you become dehydrated, your blood volume decreases, but the cellular and protein components remain the same, effectively concentrating your blood and increasing its viscosity. Picture trying to pour thick honey versus thin honey. Dehydrated blood becomes more difficult for your heart to pump, requiring greater effort to maintain the same circulation. This is why proper hydration supports cardiovascular function so effectively. When you're well hydrated, your blood maintains optimal viscosity, flowing smoothly and efficiently throughout your circulatory system. Your heart doesn't have to work as hard, and circulation to all tissues remains robust. The simple act of drinking adequate water represents one of the most direct ways you can support the physical properties that make blood an effective transport medium. The flow characteristics of blood also exhibit fascinating properties that differ significantly from simple fluids like water. Blood demonstrates what scientists call non-Newtonian flow behavior, meaning its apparent viscosity changes depending on the conditions under which it's flowing. In large vessels where blood flows rapidly, it behaves more like a thinner fluid. In smaller vessels where flow is slower, it exhibits higher apparent viscosity. This non-Newtonian behavior results primarily from the flexibility and deformability of red blood cells. In rapidly flowing blood, red blood cells tend to align with the flow direction and may even form temporary arrangements that reduce overall resistance to flow. In slower moving blood, cells tumble and interact more randomly, creating greater resistance. This represents another elegant adaptation. Blood's flow characteristics automatically adjust to the conditions within different parts of your circulatory system. The relationship between viscosity and flow also helps explain why exercise has such beneficial effects on circulation. During physical activity, increased heart rate and cardiac output create faster flow velocities throughout your circulatory system. This faster flow takes advantage of blood's non-Newtonian properties, effectively reducing its apparent viscosity and improving circulation to all tissues. The enhanced circulation during exercise helps deliver oxygen and nutrients more efficiently, while also improving the removal of metabolic waste products. Understanding blood viscosity also illuminates why certain medical conditions can significantly impact cardiovascular function. Conditions that increase red blood cell count, such as polycythemia, can dramatically increase blood viscosity, forcing the heart to work much harder to maintain circulation. Conversely, severe anemia reduces blood viscosity, and while this makes the heart's pumping job easier, it comes at the cost of reduced oxygen carrying capacity. As we prepare to explore blood's sophisticated transport functions in our next section, take a moment to appreciate these fundamental physical properties that make all of blood's biological functions possible. The careful balance of viscosity, temperature, and flow characteristics represents one of nature's most successful engineering solutions, creating a fluid that can carry tremendous diversity of cargo while maintaining the physical properties necessary for efficient circulation. Allow yourself to feel grateful for this remarkable balance that requires no conscious management from you. Your body automatically regulates blood viscosity through fluid balance, temperature control, and the production of blood cells and plasma proteins. This seamless integration of physical and biological properties ensures that the complex transport functions we've been exploring can continue effectively throughout your entire life. The knowledge that something as simple as staying well hydrated can directly support these optimal physical properties provides a gentle reminder of how everyday choices contribute to the remarkable system sustaining you. Every glass of water you drink helps maintain the perfect viscosity that allows your blood to flow smoothly, carrying oxygen, nutrients, and all the other substances that keep every cell in your body functioning optimally. Let this appreciation for blood's perfect physical properties settle peacefully in your mind as we prepare to explore the sophisticated transport network that these properties make possible. As we continue our gentle journey through the remarkable world of blood, let yourself settle even more deeply into this state of calm understanding. We now explore one of blood's most fundamental roles, serving as your body's most sophisticated delivery system. Picture blood as countless molecular delivery trucks, constantly picking up and dropping off precious cargo at precise destinations throughout your body, creating a continuous flow of essential substances that keeps every cell functioning in perfect harmony. This transport function represents one of blood's most elegant capabilities. Every moment as you rest and learn, your blood is simultaneously carrying nutrients from your digestive system to hungry cells, removing waste products that cells no longer need, and delivering chemical messages that coordinate the activities of distant organs. This constant, quiet exchange connects every part of your body in a seamless network of molecular communication and support. Let us begin by exploring how blood transports the nutrients that fuel your cellular metabolism. When you eat, your digestive system breaks down food into its component molecules: glucose from carbohydrates, amino acids from proteins, fatty acids from fats, along with vitamins and minerals from various sources. These nutrients don't simply accumulate in your digestive tract. They must be transported to the trillions of cells throughout your body that depend on them for energy, growth, and repair. Glucose, your body's primary cellular fuel, dissolves readily into plasma and circulates throughout your bloodstream. Think of glucose as molecular currency, universally accepted by virtually every cell in your body as payment for their metabolic activities. Your liver serves as a glucose bank, storing excess glucose as glycogen when supplies are abundant and releasing it back into circulation when blood glucose levels drop. This ensures that even during periods when you're not eating, your cells continue to receive the energy they need. The transport of glucose represents one of your body's most carefully regulated processes. After a meal, as glucose levels in your blood rise, your pancreas releases insulin, a hormone that acts like a molecular key, unlocking cellular doors and allowing glucose to enter cells where it can be used for energy. Between meals, when blood glucose begins to decline, your pancreas releases glucagon, which signals your liver to release stored glucose back into circulation. This delicate dance ensures that glucose availability remains steady despite the intermittent nature of eating. Amino acids, the building blocks of proteins, follow similar pathways through your bloodstream. After proteins in your food are broken down by digestion, the resulting amino acids dissolve into plasma and travel to cells throughout your body. Some cells use these amino acids to build new proteins, perhaps muscle proteins if you've been exercising, or antibodies if your immune system needs reinforcement. Others may convert amino acids into glucose if energy is needed more urgently than protein synthesis. The transport of lipids, fats, and fat-soluble substances requires more sophisticated packaging due to their water-repelling properties. Since plasma is primarily water, fats cannot simply dissolve and circulate freely. Instead, your liver packages lipids into specialized transport vehicles called lipoproteins. Think of these as molecular submarines, with a water-compatible outer shell that allows them to travel through plasma while carrying water-repelling cargo safely in their cores. Different types of lipoproteins carry different cargoes and travel to different destinations. Low-density lipoproteins, LDL, transport cholesterol and other lipids from your liver to cells that need them for membrane construction and hormone production. High-density lipoproteins, HDL, perform the reverse journey, collecting excess cholesterol from tissues and transporting it back to your liver for processing. This bidirectional lipid transport system ensures that every cell has access to the fats it needs while preventing harmful accumulation in tissues. Vitamins travel through blood, using various transport mechanisms depending on their chemical properties. Water-soluble vitamins like vitamin C and the B-complex vitamins dissolve directly into plasma and circulate freely, making them readily available to cells but also easily lost through kidney filtration if consumed in excess. Fat-soluble vitamins A, D, E, and K travel within the lipoproteins we just discussed, allowing them to be stored in fatty tissues for future use when dietary intake is low. Minerals and electrolytes each have their own specialized transport systems. Iron, essential for hemoglobin formation, travels bound to a protein called transferrin, which delivers it specifically to cells that need it for red blood cell production or other iron-requiring processes. Calcium circulates both in free ionic form and bound to plasma proteins, ready to support bone formation, muscle contraction, and nerve function. Sodium, potassium, and chloride maintain the electrical and osmotic balance necessary for cellular function while traveling freely in plasma. While blood delivers essential nutrients to cells, it simultaneously performs the equally important function of waste removal. Cellular metabolism produces various waste products that would become toxic if allowed to accumulate. Carbon dioxide, which we explored earlier, represents the most abundant metabolic waste, but cells also produce nitrogenous wastes from protein breakdown, cellular debris from normal wear and tear, and various other byproducts of biochemical processes. Urea represents the primary nitrogenous waste product in your blood. When cells break down amino acids for energy or when old proteins are recycled, the nitrogen-containing portions cannot be used for energy and must be eliminated. Your liver converts these nitrogen-containing compounds into urea, a relatively non-toxic substance that dissolves easily in plasma and travels to your kidneys for removal. This process ensures that potentially harmful nitrogen compounds never accumulate to dangerous levels in your tissues. Your kidneys serve as sophisticated filtration plants, continuously processing your entire blood volume to remove waste products while retaining essential substances. Blood flowing through kidney capillaries passes through specialized filtering structures that allow waste products like urea, creatinine, and excess electrolytes to be removed, while keeping useful substances like proteins, blood cells, and needed nutrients in circulation. This selective filtration process occurs constantly, ensuring that your blood maintains optimal composition for supporting cellular function. The liver also plays a crucial role in blood's waste transport function, serving as a processing center for various substances that need chemical modification before elimination. Bilirubin, produced when old red blood cells are broken down, travels through plasma to your liver, where it's chemically modified and prepared for elimination through bile. Many medications and other foreign substances are similarly processed by liver enzymes and either modified for elimination or converted into forms that can be safely stored or reused. Beyond transporting nutrients and wastes, blood serves as your body's communication network, carrying hormones and other chemical messengers between distant organs and tissues. This endocrine communication system allows different parts of your body to coordinate their activities despite being physically separated, creating the unified physiological responses necessary for optimal health. Hormones represent some of nature's most powerful and specific chemical messengers. Insulin, released by your pancreas, travels through blood to muscle and fat cells, instructing them to absorb glucose from circulation. Thyroid hormones, produced in your neck, circulate throughout your body, influencing the metabolic rate of virtually every cell. Cortisol, released by your adrenal glands during stress, travels through blood to multiple target tissues, preparing your body to respond to challenging situations. The specificity of hormone action depends on the presence of appropriate receptors on target cells. Think of hormones as molecular keys and cellular receptors as locks. Each hormone can only influence cells that have the correct receptor, ensuring that chemical messages reach only their intended destinations. This system allows precise communication between organs while preventing unintended effects on tissues that shouldn't respond to particular signals. Some chemical messengers work locally rather than systemically, but blood still plays important roles in their transport and distribution. Prostaglandins, produced by many different cell types, often act on nearby tissues to influence inflammation, pain sensation, or blood vessel diameter. While these substances typically don't travel far through circulation, blood flow determines how quickly they can reach their local targets and how effectively they can be cleared away once their work is complete. Blood also transports dissolved gases beyond just oxygen and carbon dioxide. Small amounts of nitrogen, normally inert, dissolve in blood and generally cause no problems. However, understanding this transport helps explain conditions like decompression sickness in deep-sea divers, where rapid pressure changes can cause dissolved nitrogen to form bubbles in blood and tissues. The transport of various substances through blood requires careful regulation to maintain optimal concentrations for cellular function. Too much glucose can damage blood vessels and organs. Too little can deprive cells of needed energy. Too much sodium can disrupt cellular function and blood pressure. Too little can cause dangerous muscle and nerve problems. Your body maintains elaborate feedback systems that continuously monitor blood composition and adjust the transport, production, and elimination of various substances to maintain optimal balance. As we prepare to explore blood's role in temperature regulation, take a moment to appreciate this sophisticated transport network operating within you right now. Every heartbeat circulates this molecular delivery system, ensuring that nutrients reach hungry cells, wastes are removed before they can cause harm, and chemical messages coordinate the activities of your organs and tissues. This process continues whether you're awake or asleep, active or at rest, representing one of your body's most fundamental and essential services. Allow yourself to feel grateful for this constant, quiet exchange that requires no conscious thought or management from you. The nutrients from your last meal are being delivered precisely where they're needed, while metabolic wastes are being efficiently removed. Hormones are carrying messages between organs, coordinating responses that keep your internal environment stable and optimal for life. This remarkable transport network ensures that every cell in your body remains connected to the resources it needs and the communication systems that coordinate your physiological functions. Let this appreciation for blood's sophisticated delivery system settle peacefully in your mind as we prepare to explore how this same remarkable fluid helps maintain your body temperature within the narrow range necessary for optimal cellular function. As we continue our peaceful journey through the remarkable world of blood, let yourself breathe even more slowly and deeply as we explore one of blood's most essential yet often overlooked functions, serving as your body's internal thermostat. This warming exploration reveals how blood continuously maintains your body temperature within the narrow range necessary for optimal cellular function, creating a stable thermal environment that allows the complex biochemical processes of life to continue regardless of external conditions. Picture blood as a liquid heating and cooling system, constantly flowing through your body like warm rivers, carrying thermal energy from areas where heat is produced to areas where it's needed or can be released. This isn't a simple process of moving warm liquid around. It represents one of nature's most sophisticated temperature regulation systems, operating automatically and precisely to maintain the thermal stability that complex life requires. Your body generates heat continuously through cellular metabolism. Every chemical reaction that occurs within your cells, the breakdown of glucose for energy, the synthesis of proteins, the contraction of muscles, produces heat as a byproduct. This metabolic heat production is particularly intense in organs like your liver, which performs hundreds of different chemical processes simultaneously, and your skeletal muscles, especially during physical activity. Without an effective way to distribute this heat, these metabolically active organs would overheat while other parts of your body remained too cool for optimal function. Blood solves this thermal challenge through its remarkable heat capacity, its ability to absorb and transport significant amounts of thermal energy without dramatic temperature changes. Water, which makes up the majority of plasma, has one of the highest heat capacities of any naturally occurring substance. This means that blood can absorb considerable amounts of heat from metabolically active organs without becoming dangerously hot itself. then transport this thermal energy throughout your body where it can warm cooler tissues or be released to the environment. As blood flows through your liver, your heart muscle, and your working skeletal muscles, it absorbs the excess heat generated by their intense metabolic activities. Think of blood as thermal sponges, soaking up this heat and preventing these vital organs from overheating. This heated blood then travels through your circulatory system, gradually releasing its thermal energy to cooler parts of your body, ensuring that your core temperature remains stable even when different organs are working at vastly different metabolic rates. The distribution of this absorbed heat isn't random. Your circulatory system can direct more warm blood to areas that need heating and redirect it away from areas that need cooling. When you're in a cold environment, blood vessels near your skin's surface constrict, redirecting warm blood away from areas where heat would be lost to the environment and keeping it circulating through your core organs where warmth is most needed. This vasoconstriction is why your fingers, toes, and nose can feel cold in winter weather. Blood flow to these extremities is reduced to conserve core temperature. Conversely, when you need to release heat, perhaps after exercise or in a warm environment, blood vessels near your skin surface dilate, allowing more warm blood to flow close to your body's surface, where heat can be transferred to the cooler air around you. This vasodilation is why your skin may appear flushed and feel warm when you're overheated. Increased blood flow to surface vessels brings more thermal energy close to your skin where it can dissipate into the environment. This vascular control represents one of your body's most immediate and effective temperature regulation mechanisms. The smooth muscle cells in your blood vessel walls can contract or relax within seconds in response to thermal needs, instantly redirecting blood flow to optimize heat distribution throughout your body. This process happens automatically through neural and hormonal controls that sense temperature changes and adjust circulation accordingly, requiring no conscious thought or effort from you. The thermal properties of blood itself contribute to its effectiveness as a heat distribution system. Blood's temperature typically runs about 1 degree warmer than your core body temperature, maintaining approximately 38 degrees Celsius, 100.4 degrees Fahrenheit. This slight elevation results from the continuous absorption of metabolic heat as blood passes through active organs, combined with the gentle friction generated as blood flows through your vast network of blood vessels. This elevated temperature means that blood serves as a continuous source of warmth for cooler parts of your body. As warm blood flows through less metabolically active tissues, perhaps your resting muscles, or the outer layers of your skin during mild weather, it gradually releases thermal energy, gently warming these areas to maintain optimal cellular function throughout your body. Blood's role in temperature regulation becomes particularly important during physical activity when heat production can increase dramatically. During exercise, your working muscles may generate heat at rates many times higher than at rest. Without effective heat removal, muscle temperature could rise to levels that would damage cellular proteins and disrupt normal function. Blood flowing through these active muscles absorbs this excess heat, preventing dangerous overheating while simultaneously distributing the thermal energy to support enhanced circulation and cellular activity throughout your body. The cardiovascular adjustments that occur during exercise demonstrate the elegant integration of blood's transport and thermal regulation functions. As your heart rate increases and cardiac output rises, more blood flows through your working muscles per minute, providing enhanced removal of metabolic heat along with increased delivery of oxygen and nutrients. Simultaneously, blood flow to your skin increases, allowing more efficient heat dissipation to prevent dangerous elevation of core temperature. Sweating represents another way that blood supports temperature regulation, though this involves the loss of plasma rather than the transport of heat. When your body needs to release large amounts of heat quickly, your sweat glands extract water and electrolytes from blood plasma and secrete them onto your skin surface. As this sweat evaporates, it removes significant amounts of thermal energy from your body. The plasma lost through sweating is gradually replaced through fluid intake and retention, but this process demonstrates how blood's components can be utilized for emergency cooling when other mechanisms aren't sufficient. Blood's temperature regulation function also helps explain why fever can be beneficial during illness, despite feeling uncomfortable. When your immune system detects infection, it releases substances called pyrogens that reset your body's thermal set point to a higher temperature. Blood then distributes heat more efficiently throughout your body, raising core temperature to levels that enhance immune function and create a less hospitable environment for many pathogens. The increased temperature accelerates many immune processes while potentially slowing bacterial reproduction. The thermal regulation provided by blood becomes crucial during medical procedures or conditions that might disrupt normal temperature control. During surgery, when patients are anesthetized and unable to regulate temperature normally, warming blankets and heated intravenous fluids help maintain core temperature by supporting blood's natural thermal distribution functions. In conditions like hypothermia, rewarming protocols focus on gradually increasing blood temperature to safely restore normal thermal regulation. Understanding blood's thermal regulation role also illuminates why certain cardiovascular conditions can affect temperature tolerance. When circulation is impaired, blood's ability to distribute heat effectively throughout the body becomes compromised. This explains why people with certain heart conditions or circulation problems may have difficulty tolerating temperature extremes. Their blood can't efficiently redistribute thermal energy to maintain comfortable core and peripheral temperatures. As we prepare to explore how blood pressure drives all the circulation we've been discussing, take a moment to appreciate this thermal regulation system working within you right now. Blood is continuously absorbing heat from your most metabolically active organs, distributing this thermal energy throughout your body, and adjusting its flow patterns to maintain optimal temperature in all your tissues. This process continues whether you're in a warm room or cool air, whether you're resting quietly or being physically active. Allow yourself to feel grateful for this automatic climate control system that maintains the stable thermal environment necessary for optimal cellular function. Every heartbeat circulates this temperature-regulating fluid, ensuring that biochemical processes throughout your body can proceed at their optimal rates regardless of external conditions or internal variations in heat production. This thermal stability represents one of the fundamental requirements for complex life, made possible by blood's remarkable thermal properties and your circulatory system's ability to direct its flow precisely where thermal regulation is needed most. Let this appreciation for blood's role as your internal thermostat settle peacefully in your mind as we prepare to explore the gentle but persistent pressure that drives all the circulation, making these vital functions possible. As we continue our peaceful journey through the remarkable world of blood, let yourself settle even more deeply into this state of calm learning. We now explore the gentle but persistent force that drives all the circulation we've been discovering together: blood pressure. This rhythmic pressure represents the mechanical foundation upon which all of blood's transport, protective and regulatory functions depend. Picture your heart as a tireless, gentle pump, creating waves of pressure that ripple through your entire circulatory system like a stone dropped into a calm pond. These pressure waves, generated with each heartbeat, propel blood through an intricate network of vessels that reaches every cell in your body. Right now, as you rest quietly, your heart is creating approximately 60 to 100 of these pressure waves each minute, each one ensuring that oxygen, nutrients, and all the vital substances we've explored continue their essential journeys. Blood pressure isn't a single constant force. It's a rhythmic, pulsating pressure that changes with each cardiac cycle. When your heart contracts during systole, it creates the peak pressure that pushes blood forward through your arteries. This systolic pressure represents the maximum force generated by your heart's muscular contraction, typically ranging from 90 to 140 millimeters of mercury in healthy adults. Think of this as the driving force that initiates each cycle of circulation, providing the energy needed to move blood from your heart toward the distant capillaries in your tissues. Between heartbeats, during diastole, your heart relaxes and refills with blood returning from your body. During this relaxation phase, pressure in your arteries doesn't drop to zero. Instead, it maintains what we call diastolic pressure, typically ranging from 60 to 90 millimeters of mercury. This sustained pressure between heartbeats ensures continuous blood flow even when your heart isn't actively contracting. The elastic walls of your larger arteries, stretched during systole, gently recoil during diastole, helping to maintain forward flow and preventing circulation from stopping between heartbeats. This alternating pattern of higher and lower pressure creates the pulse you can feel at various points on your body, your wrist, your neck, your temple. Each pulse represents one complete cardiac cycle, one wave of pressure traveling from your heart through your arterial system. The pulse you feel isn't actually blood moving under your finger, but rather the pressure wave expanding the artery wall as it passes. This pressure wave travels much faster than the blood itself, reaching your extremities almost instantaneously, while the blood takes much longer to complete its journey. Blood pressure varies dramatically throughout your circulatory system, creating the pressure gradient that ensures steady directional flow. Pressure is highest in your aorta, the large artery that carries blood directly from your heart, where systolic pressures may reach 120 millimeters of mercury or higher. As blood moves through progressively smaller arteries, arterioles, and capillaries, pressure gradually decreases, reaching very low levels in your venous system where blood returns to your heart. This pressure gradient is essential for proper circulation. Just as water flows downhill from areas of high pressure to areas of low pressure, blood flows from the high-pressure arterial system through the intermediate pressure capillary beds to the low-pressure venous system. Without adequate pressure differences, blood would not flow consistently in the proper direction, and the transport functions we've explored would become ineffective. The capillary beds represent a particularly important region where pressure plays a crucial role in the exchange of materials between blood and tissues. Capillary pressure must be high enough to drive the filtration of plasma and dissolved substances out of blood vessels and into tissue spaces, where cells can access nutrients and oxygen. Simultaneously, it must not be so high as to cause excessive fluid loss or damage to delicate capillary walls. This delicate balance allows the efficient exchange of materials while maintaining proper fluid distribution between your circulatory system and your tissues. As blood enters your venous system, pressure drops to very low levels, sometimes only a few millimeters of mercury. This creates a challenge. How does blood return to your heart against gravity, particularly from your lower extremities? Your body solves this challenge through several elegant mechanisms that work together to ensure adequate venous return despite low driving pressure. Venous valves, one-way structures within your veins, prevent blood from flowing backward due to gravity. These valves open when blood flows toward your heart and close when blood tries to flow in the opposite direction. Think of them as molecular turnstiles, ensuring that blood can only travel in the proper direction through your venous system. The muscle pump mechanism utilizes the contractions of your skeletal muscles to assist venous return. When muscles contract during movement, they compress nearby veins, pushing blood toward your heart. When muscles relax, venous valves prevent the blood from flowing backward, creating a net forward movement despite the low pressure in venous blood. This is why prolonged inactivity can sometimes lead to pooling of blood in your lower extremities, while gentle movement promotes healthy circulation. Your respiratory pump also assists venous return through the pressure changes that occur during breathing. When you inhale, pressure in your chest cavity decreases, creating a gentle suction that helps draw venous blood back to your heart. When you exhale, this pressure gradient reverses, but venous valves prevent backward flow. This means that each breath you take contributes subtly to the circulation of blood throughout your body. Blood pressure regulation involves sophisticated feedback systems that continuously monitor and adjust pressure levels to meet your body's changing needs. Specialized pressure sensors called baroreceptors, located in your major arteries, constantly monitor blood pressure and send information to cardiovascular control centers in your brain. When these sensors detect pressure changes, they trigger automatic adjustments in heart rate, the force of cardiac contraction, and the diameter of blood vessels to maintain optimal pressure for current conditions. During physical activity, your cardiovascular system automatically increases blood pressure to meet the enhanced circulation needs of working muscles. Your heart rate increases, your heart contracts more forcefully, and blood vessels in active muscles dilate to accommodate increased flow. These adjustments happen without conscious thought, ensuring that increased metabolic demands are met with appropriately enhanced circulation. When you're at rest, particularly during sleep, your cardiovascular system reduces blood pressure to levels appropriate for maintenance functions. Heart rate slows, cardiac output decreases, and overall pressure drops to conserve energy while still maintaining adequate circulation for essential physiological processes. This nocturnal reduction in blood pressure allows your cardiovascular system to rest and recover while continuing to meet your body's basic needs. The relationship between blood pressure and circulation becomes particularly evident during postural changes. When you stand up quickly, gravity causes blood to pool temporarily in your lower extremities, reducing the volume returning to your heart and potentially causing a brief drop in blood pressure. Your baroreceptor system responds within seconds, increasing heart rate and causing blood vessel constriction to restore adequate pressure and prevent dizziness or fainting. Blood vessel diameter plays a crucial role in pressure regulation through its effect on resistance to flow. Small changes in the diameter of arterioles, the smallest arteries, can have dramatic effects on blood pressure and flow distribution. When arterioles constrict, resistance increases and blood pressure rises. When they dilate, resistance decreases and pressure drops. This allows your body to fine-tune circulation to specific organs and tissues based on their current needs. The kidneys play a particularly important role in long-term blood pressure regulation through their control of blood volume. When blood pressure is too high, your kidneys can increase fluid excretion, reducing blood volume and thereby reducing pressure. When pressure is too low, they can retain more fluid, increasing blood volume and pressure. This volume-based pressure control works more slowly than neural mechanisms, but provides essential long-term stability for your circulatory system. As we prepare to explore the molecular markers that make each person's blood unique, take a moment to appreciate this rhythmic driving force working within you right now. Every heartbeat creates a pressure wave that ensures the continued circulation of all the remarkable components we've been exploring. The oxygen-carrying red blood cells, the protective white blood cells, the healing platelets, and the nutrient-rich plasma that sustains every cell in your body. Allow yourself to feel grateful for this persistent, gentle pressure that requires no conscious management from you. Your cardiovascular system automatically adjusts this driving force based on your activities, your position, and your body's changing needs, ensuring that circulation remains optimal whether you're active or at rest, awake or asleep. This mechanical foundation makes possible all the chemical, biological, and thermal functions we've discovered, creating the continuous flow that connects every part of your body in a unified living system. The rhythmic nature of blood pressure, the gentle rise and fall with each heartbeat, represents one of life's most fundamental rhythms. This pressure rhythm has been sustaining you since before your birth and will continue throughout your entire life, a persistent reminder of the mechanical processes that support the remarkable biological complexity of your circulatory system. Let this appreciation for the rhythm of life flowing through your vessels settle peacefully in your mind as we prepare to explore the molecular signatures that make your blood uniquely yours. As we continue our peaceful journey through the remarkable world of blood, let yourself breathe even more slowly and deeply as we explore one of nature's most fascinating examples of molecular recognition, blood types. These microscopic markers displayed on the surface of your red blood cells like molecular name tags create a unique identity for your blood that has profound implications for medical care and our understanding of human diversity. Think of blood types as a sophisticated molecular language, a system of chemical markers that allows your immune system to distinguish between self and foreign with extraordinary precision. Right now, as you rest quietly, every red blood cell flowing through your vessels carries specific molecular signatures that identify them as belonging uniquely to you. These markers represent one of your body's most fundamental recognition systems, determining how your blood will interact with blood from other individuals. The story of blood types begins with the ABO system, discovered over a century ago and representing one of medicine's most important breakthroughs. This system is based on the presence or absence of specific sugar molecules, called antigens, attached to the surface of red blood cells. These antigens aren't random decorations. They're precise molecular structures that your immune system has learned to recognize as part of your own cellular identity. If you have type A blood, your red blood cells display antigens on their surfaces, specific sugar molecules that create a unique molecular signature. Your immune system recognizes these antigens as self and will not attack red blood cells that carry them. However, this same immune system has learned to recognize B antigens, which you don't possess, as foreign and potentially dangerous. This recognition system developed early in your life as your immune system catalogued the molecular markers that belong to your body. Type B blood carries the opposite pattern, B antigens on red blood cell surfaces, with an immune system trained to accept B antigens as self while treating antigens as foreign invaders. Type B blood presents both A and B antigens simultaneously, creating red blood cells that carry both molecular signatures. The immune systems of people with AB blood recognize both A and B antigens as self, making them universal recipients for red blood cell transfusions from the ABO perspective. Type O blood represents a unique situation, red blood cells that lack both A and B antigens entirely. Think of type O red blood cells as having clean surfaces, unmarked by either of the major ABO antigens. This absence of antigens means that type O blood won't trigger immune responses in people with any ABO blood type, earning type O individuals the designation of universal donors for red blood cell transfusions. The rouge system adds another layer of complexity to blood typing, based on the presence or absence of a different set of antigens, most importantly the rouge day antigen. If your red blood cells carry the rouge day antigen, you're considered rouge positive. If they lack this antigen, you're rouge negative. This creates 8 common blood types when combined with the ABO system: A+, AB+, BAB+, ABO+, and O-MO. The molecular basis of these blood type systems reflects the incredible precision of cellular recognition. The A and B antigens differ by only a single sugar molecule, galactosin B antigens versus N-acetyl-galactosamine in A antigens. This tiny molecular difference is sufficient for your immune system to distinguish between them with absolute reliability, demonstrating the exquisite sensitivity of biological recognition systems. Your blood type is determined by genes inherited from your parents, following predictable patterns that geneticists have mapped in detail. The ABO gene exists in 3 main versions: A, B, and O, with A and B being dominant over O. This means that receiving an A gene from one parent and an O gene from the other results in type A blood, while receiving O genes from both parents results in type O blood. The Rive system follows similar inheritance patterns, with Rive positive being dominant over Rive negative. Understanding blood type compatibility becomes crucial for safe blood transfusions, representing one of medicine's most critical applications of immunological knowledge. When blood types are incompatible, the recipient's immune system recognizes the donor's red blood cells as foreign invaders and launches an attack called a hemolytic transfusion reaction. This immune response can destroy the transfused red blood cells, potentially causing serious complications including kidney damage, shock, and even death. The compatibility rules follow the recognition patterns we've discussed. Type O negative blood, lacking both ABO antigens and the rouge day antigen, can be safely transfused to anyone in emergency situations, making O negative donors particularly valuable for emergency medicine. Type AB positive blood, carrying all the major antigens, means that people with this blood type can receive red blood cells from any ABO or type, though they can only donate to other AB positive recipients. Beyond the major ABO and rouge systems, scientists have identified hundreds of other blood group systems, each based on different molecular markers present on red blood cell surfaces. Systems like Duffy, Kidd, Lewis, and MNS represent additional layers of molecular diversity that can become important in specific medical situations, particularly for patients who require multiple transfusions over time. The plasma component of blood compatibility follows opposite rules from red blood cell compatibility. While type O individuals can donate red blood cells to anyone, their plasma contains antibodies against both A and B antigens, making type O plasma compatible only with other type O recipients. Conversely, type OB plasma lacks antibodies against A or B antigens, making it compatible with recipients of any ABO type. This antibody pattern in plasma develops naturally during early life through exposure to environmental substances that resemble blood-type antigens. Your immune system encounters A-like or B-like molecules in food, bacteria, and other environmental sources, and if these molecules differ from your own blood-type antigens, your immune system produces antibodies against them. This natural antibody production ensures that by adulthood, your plasma contains antibodies against any ABO antigens you don't possess on your own red blood cells. Blood typing has revolutionized not only transfusion medicine, but also organ transplantation, where compatibility extends beyond blood types to include tissue compatibility markers called HLA, human leukocyte antigens. These systems work together to determine whether transplanted organs will be accepted or rejected by recipient immune systems, highlighting the broader importance of molecular recognition in modern medicine. The distribution of blood types varies among different populations worldwide, reflecting human migration patterns and evolutionary history. Type O blood is most common globally, found in about 45% of people, while AB blood is relatively rare at about 4% of the population. These frequency differences have important implications for blood banking and medical planning in different regions of the world. Recent research has revealed that blood types may influence susceptibility to certain diseases and health conditions, though the mechanisms aren't always fully understood. Some studies suggest that certain blood types may be associated with different risks for cardiovascular disease, certain infections or blood clotting disorders, though these associations represent statistical trends rather than individual predictions. As we prepare to explore what happens when blood's delicate systems need medical support, take a moment to appreciate this remarkable molecular recognition system operating within you right now. Every red blood cell in your circulation carries the specific molecular markers that identify it as uniquely yours, while your immune system maintains constant vigilance to ensure that only compatible blood components remain in your system. Allow yourself to feel grateful for this sophisticated identification system that enables safe medical care when blood transfusions become necessary. The molecular markers on your red blood cells represent a biological signature as unique as your fingerprints, yet compatible with others who share similar molecular patterns. This system of recognition and compatibility has made possible countless medical interventions that save lives every day, from emergency transfusions to complex surgical procedures. The concept that your blood carries molecular information about your genetic heritage adds another layer of wonder to the fluid we've been exploring together. These blood type markers connect you to your ancestors while also determining your compatibility with potential blood donors from around the world, creating a global network of molecular compatibility that transcends all other human differences. Let this appreciation for your blood's unique molecular signature settle peacefully in your mind as we prepare to explore how medical science supports blood systems when they need assistance. As we continue our gentle journey through the remarkable world of blood, let yourself settle even more deeply into this peaceful state of learning. We now turn our attention to what happens when blood's delicate systems encounter challenges that require medical support. In this compassionate section, we'll softly examine common blood disorders, not to create worry or anxiety, but to deepen our appreciation for blood's normal, healthy function, and to understand how medical science helps restore balance when it's needed. Think of blood disorders as disruptions in the elegant harmony we've been exploring together. Just as a symphony occasionally requires the conductor to adjust tempo, or bring certain instruments back into balance. Your blood systems sometimes need medical guidance to restore their optimal function. These conditions affect millions of people worldwide, and understanding them helps us appreciate both the complexity of normal blood function and the remarkable advances in medical care that help people live healthy, fulfilling lives even when their blood systems need support. Let us begin by gently exploring anemia, a condition where the blood's oxygen carrying capacity becomes reduced below optimal levels. Picture anemia as your circulatory system running with fewer delivery trucks than needed. The transport network continues to function, but it cannot carry as much oxygen as your tissues require for optimal energy production. This condition affects hundreds of millions of people globally, yet with proper understanding and care, most forms of anemia can be effectively managed. The most common form, iron deficiency anemia, occurs when your body lacks sufficient iron to produce adequate amounts of healthy hemoglobin. Remember our earlier exploration of hemoglobin, that remarkable protein within red blood cells that carries oxygen through iron atoms at its core. When iron supplies become insufficient, your bone marrow cannot manufacture enough functional hemoglobin, resulting in red blood cells that are smaller than normal and carry less oxygen than they should. This condition often develops gradually, and your body initially compensates remarkably well. Your heart may beat slightly faster to circulate the available oxygen carrying capacity more quickly, and your breathing may become slightly more efficient. Many people with mild iron deficiency anemia continue their daily activities normally, though they may notice increased fatigue, especially during physical activity when oxygen demands are higher. The gentle treatment for iron deficiency anemia often involves iron supplementation combined with dietary modifications to include iron-rich foods and substances that enhance iron absorption. Your body responds to these interventions by gradually increasing hemoglobin production, restoring oxygen carrying capacity over a period of weeks to months. This represents a beautiful example of how supporting your body's natural processes can restore optimal function when temporary imbalances occur. We'll softly explore other forms of anemia as well, including those caused by vitamin deficiencies, chronic diseases, or genetic factors. Vitamin B12 and folate deficiencies can prevent your bone marrow from producing normal red blood cells, resulting in larger but less effective cells that cannot carry oxygen efficiently. Chronic kidney disease can reduce production of erythropoietin, the hormone that stimulates red blood cell production, while certain autoimmune conditions can interfere with iron absorption or utilization. Let us now turn our attention to conditions affecting the white blood cells we met earlier, those silent guardians that protect your health. Leukemia represents a group of conditions where abnormal white blood cells multiply excessively, disrupting the normal balance within your blood and bone marrow. Think of leukemia as a miscommunication within your blood cell production system, where the normal controls that regulate cell division and maturation become disrupted. Rather than focusing on the challenges these conditions present, we'll gently explore how modern medicine has developed sophisticated treatments that work with your body's natural healing capabilities. Many forms of leukemia now respond well to targeted therapies that specifically address the cellular miscommunications causing abnormal cell growth, while preserving normal blood cell production as much as possible. This section will softly discuss how treatments like chemotherapy, immunotherapy, and stem cell transplantation represent ways of resetting your blood cell production system, allowing healthy stem cells to re-establish normal blood cell manufacturing. These treatments often achieve remarkable success, with many people returning to completely normal lives after treatment completion. We'll explore clotting disorders with the same gentle, hopeful perspective, understanding conditions where the elegant three-step hemostasis process we explored earlier becomes imbalanced. Some conditions, like hemophilia, affect the coagulation cascade we discovered, preventing normal clot formation when blood vessels are injured. Other conditions create the opposite problem, with excessive clotting that can block blood vessels inappropriately. Hemophilia beautifully illustrates how understanding blood's normal function leads to effective treatments. When specific clotting factors are deficient or absent, medical science can provide factor replacement therapy, essentially supplying the missing molecular tools that enable normal clot formation. People with hemophilia can live active, normal lives when they have access to appropriate factor replacement, demonstrating how supporting natural processes can overcome genetic variations. The section will gently explore how conditions affecting platelet function or number can be managed through various approaches, from medications that support platelet production to treatments that help platelets function more effectively. We'll discover how understanding the normal role of platelets, those gentle healers we met earlier, has led to treatments that preserve their essential functions while addressing any imbalances. Throughout this exploration, we'll emphasize how blood disorders have driven many of medicine's most important advances. The need to understand and treat anemia led to the discovery of vitamins and their roles in cellular metabolism. Research into leukemia contributed to the development of targeted cancer therapies that benefit people with many different conditions. Studies of clotting disorders have resulted in both treatments for bleeding problems and medications that prevent dangerous blood clots. We'll softly discuss how modern diagnostic capabilities allow healthcare providers to identify blood disorders early, often before symptoms become severe. Simple blood tests can reveal changes in cell counts, hemoglobin levels, and clotting function that indicate when systems need support. This early detection enables gentle interventions that can prevent more serious complications while supporting your body's natural healing and adaptation capabilities. The section will explore how lifestyle factors can support healthy blood function, from adequate nutrition that provides the building blocks for blood cell production to regular physical activity that promotes healthy circulation. We'll discover how staying well hydrated supports optimal blood viscosity, while adequate rest allows your bone marrow to maintain healthy blood cell production cycles. As we prepare to explore how healthcare providers read blood's story through laboratory testing, take a moment to appreciate the resilience and adaptability of the blood systems we've been exploring together. Even when challenges arise, your body's natural healing capabilities, supported by modern medical understanding, can often restore optimal function and maintain the remarkable processes that sustain life. Allow yourself to feel hopeful about the continuous advances in medical science that help people whose blood systems need support. Every condition we've gently explored has inspired research that has led to better treatments, and this progress continues every day. The same curiosity that led to our understanding of normal blood function drives ongoing efforts to help people maintain healthy, active lives, even when their blood systems require medical assistance. The knowledge that blood disorders affect people from all walks of life, yet many can be effectively managed or treated, should inspire gratitude for both the resilience of human biology and the dedication of medical professionals who work to understand and support these vital systems. This understanding helps us appreciate not only the remarkable complexity of normal blood function, but also the ways that medical science can partner with your body's natural healing capabilities to maintain health and vitality. Let this appreciation for both the complexity of blood disorders and the hope offered by modern medical care settle peacefully in your mind as we prepare to explore how simple laboratory tests can reveal the remarkable story your blood tells about your health. As we continue our peaceful journey through the remarkable world of blood, let yourself breathe even more slowly and deeply as we explore one of medicine's most valuable diagnostic tools, laboratory testing, that reveals the intimate story your blood tells about your health and well-being. In this gentle conclusion to our technical exploration, we'll softly discover how healthcare providers read blood's story through careful analysis, transforming small samples into detailed conversations with your body's internal systems. Think of blood tests as a detailed conversation with your body, where each measurement provides insight into the quiet processes we've been exploring together. When you provide a blood sample for laboratory analysis, you're essentially offering a snapshot of your internal environment, a liquid biopsy that contains information about virtually every organ system and metabolic process occurring within you at that moment. This remarkable capability transforms blood into a diagnostic treasure trove, allowing healthcare providers to assess your health, monitor treatment progress, and detect potential problems before they become serious. The most common and comprehensive blood test is the complete blood count, affectionately known as the CBC. This foundational test examines the cellular components of blood we've met throughout our journey: red blood cells, white blood cells, and platelets, counting them, measuring their characteristics, and assessing their overall health and function. Picture the CBC as taking an inventory of your blood's cellular residents, ensuring that each population is present in appropriate numbers and displaying normal characteristics. The red blood cell portion of the CBC provides multiple measurements that reflect your oxygen carrying capacity and overall blood health. The red blood cell count tells us how many of these oxygen carriers are present in each microliter of your blood, typically ranging from 4.5 to 5.5 million cells per microliter for men and 4.0 to 5.0 million for women. Don't worry about memorizing these exact numbers. What's important is understanding that these counts reflect your bone marrow's production of these essential oxygen transporters. Hemoglobin levels, measured in grams per deciliter, indicate the actual oxygen carrying capacity of your blood, regardless of cell count. Remember our exploration of hemoglobin, that iron-rich protein that gives blood its red color and enables oxygen transport. Normal hemoglobin levels typically range from 14 to 18 grams per deciliter for men and 12 to 16 grams per deciliter for women, reflecting the total amount of this crucial protein available for oxygen transport. Hematocrit represents the percentage of your blood volume occupied by red blood cells, providing insight into the proportion of cellular components versus plasma in your circulation. Picture hematocrit as measuring how much of your blood consists of those biconcave oxygen carriers we explored earlier versus the golden plasma that serves as their transport medium. Normal hematocrit values typically range from 41% to 50% for men and 36% to 44% for women. The CBC also examines red blood cell characteristics through measurements called indices, which provide information about cell size, hemoglobin content, and overall cell health. Mean corpuscular volume, MCV, measures the average size of red blood cells, helping to identify whether cells are larger or smaller than normal. Mean corpuscular hemoglobin, MCH, and mean corpuscular hemoglobin concentration, MCHC, assess how much hemoglobin each cell contains, providing insights into iron status and hemoglobin production efficiency. White blood cell analysis through the CBC includes both total white blood cell count and a differential count that breaks down the percentages of different white blood cell types. The total count, typically ranging from 4,000 to 11,000 cells per microliter, provides an overall assessment of immune system activity. The differential count examines the percentages of neutrophils, lymphocytes, monocytes, eosinophils, and basophils, those protective guardians we met earlier in our journey. Neutrophil percentages, normally comprising 50 to 70% of white blood cells, can indicate bacterial infections when elevated or certain bone marrow problems when decreased. Lymphocyte percentages, typically 20 to 30% of white blood cells, may rise during viral infections or certain immune system conditions. The percentages of other white blood cell types provide additional insights into immune function, allergic responses, and various disease processes. Platelet analysis completes the cellular portion of the CBC, counting those tiny cellular fragments we discovered as gentle healers in our earlier exploration. Normal platelet counts typically range from 150,000 to 400,000 per microliter, ensuring adequate numbers of these repair specialists are available for hemostasis when needed. The CBC may also provide information about platelet size and characteristics, offering insights into platelet production and function. Beyond the CBC, comprehensive metabolic panels examine the chemical composition of your blood plasma, revealing information about organ function, metabolism, and chemical balance. These tests analyze the dissolved substances we explored earlier: glucose, electrolytes, proteins, and waste products, providing insights into how well your organs are performing their essential functions. Blood glucose levels, typically measured after fasting, provide information about your body's ability to regulate blood sugar through the insulin and glucagon mechanisms we discussed earlier. Normal fasting glucose levels usually range from 70 to 100 milligrams per deciliter, reflecting optimal balance between glucose production, consumption, and storage. Elevated levels may indicate problems with insulin production or function, while low levels might suggest issues with glucose production or excessive consumption. Electrolyte measurements examine the sodium, potassium, chloride, and carbon dioxide levels that maintain proper cellular function and acid-base balance. These measurements reflect kidney function, fluid balance, and the body's ability to maintain the precise chemical environment necessary for optimal cellular activity. Remember our exploration of how electrolytes maintain the electrical properties necessary for nerve conduction and muscle contraction. These laboratory values assess whether these critical balances are being maintained properly. Kidney function assessment through blood tests examines waste products like blood, urea, nitrogen, BUN, and creatinine that accumulate when kidney filtration becomes impaired. These measurements reflect how effectively your kidneys are performing the waste removal functions we explored earlier, when we discovered how blood carries cellular waste products to organs capable of elimination. Liver function tests examine enzymes and proteins produced by your liver, providing insights into this crucial organ's ability to process the substances carried by blood. Tests like alanine aminotransferase, ALT, and aspartate aminotransferase, AST, measure enzymes that leak from liver cells when they're damaged, while albumin and total protein levels reflect the liver's synthetic functions we discussed when exploring plasma proteins. Lipid panels analyze the cholesterol and triglyceride levels in your blood, examining the lipoproteins we discovered earlier that transport fats through your aqueous plasma. These tests provide insights into cardiovascular risk by assessing the balance between different types of cholesterol transporters and overall lipid metabolism. Thyroid function tests examine hormone levels that reflect the activity of your thyroid gland, including thyroid-stimulating hormone TSH and thyroid hormones T3 and T4. Remember our exploration of how blood serves as a communication network, carrying hormones between organs. These tests assess whether thyroid hormone communication is functioning optimally. Inflammatory markers like C-reactive protein, CRP, and erythrocyte sedimentation rate, ESR, provide information about inflammatory processes occurring throughout your body. These tests don't identify specific conditions, but rather indicate whether your immune system is responding to injury, infection, or other inflammatory stimuli. Coagulation studies assess the clotting mechanisms we explored in our section on hemostasis, measuring how quickly and effectively your blood can form clots when needed. Tests like prothrombin time PT and partial thromboplast in time PTT evaluate different aspects of the coagulation cascade, ensuring that the elegant three-step process of hemostasis is functioning properly. Specialized tests can examine specific aspects of blood function in greater detail when needed. Vitamin B12 and folate levels assess whether adequate nutrients are available for healthy red blood cell production. Iron studies examine iron stores, transport, and utilization, providing detailed insights into iron deficiency, anemia, or iron overload conditions. Hemoglobin electrophoresis can identify genetic variations in hemoglobin structure that affect oxygen transport. The beauty of laboratory testing lies in how these individual measurements combine to tell comprehensive stories about health and disease. A healthcare provider doesn't typically look at single values in isolation, but rather examines patterns and relationships between different measurements to understand what your blood is revealing about your overall health status. As we prepare to explore how blood changes throughout the human lifespan, take a moment to appreciate how these laboratory measurements transform the invisible processes we've been exploring into concrete, measurable information. The same red blood cells, white blood cells, platelets, and plasma components we've met throughout our journey become quantifiable data that can guide medical decisions and monitor health over time. Allow yourself to feel grateful for the sophisticated analytical capabilities that can transform a small blood sample into detailed information about the remarkable systems sustaining your life. These measurements represent windows into the cellular and molecular processes occurring within you, providing healthcare providers with the information needed to support your health and detect problems before they become serious. The concept that your blood tells such a complete story about your internal environment adds another layer of wonder to the fluid we've been exploring together. Every laboratory value reflects the activities of specific organs, the balance of metabolic processes, and the overall harmony of the systems that maintain your health and vitality. Let this appreciation for blood's diagnostic capabilities settle peacefully in your mind as we prepare to explore how this remarkable system changes and adapts throughout the human lifespan, from birth to maturity. As our peaceful journey through the remarkable world of blood nears its gentle conclusion, let yourself settle even more deeply into this state of calm understanding. We now explore one of the most beautiful aspects of blood, how it changes, adapts, and evolves throughout the human lifespan. From the moment of conception until the final stages of life, blood demonstrates an extraordinary capacity for adaptation, meeting the changing needs of growth, development, and aging with remarkable flexibility and wisdom. The story of blood throughout life begins even before birth, in the earliest stages of human development. During those quiet weeks in the womb, blood takes on characteristics perfectly suited for life before breathing begins. Fetal blood differs fundamentally from adult blood, containing specialized hemoglobin that has a higher affinity for oxygen than the hemoglobin we explored earlier. Think of fetal hemoglobin as being designed with a stronger grip on oxygen molecules, ensuring efficient transfer across the placenta from maternal circulation to the developing baby. This fetal hemoglobin, designated HEP, represents nature's perfect adaptation for intrauterine life. While adult hemoglobin readily releases oxygen when it encounters the slightly acidic, carbon dioxide-rich environment of working tissues, fetal hemoglobin holds onto oxygen more tenaciously. This characteristic ensures that even in the lower oxygen environment of the womb, the developing baby's tissues receive adequate oxygen for the rapid growth and development occurring during these crucial months. The transition from fetal to adult hemoglobin begins even before birth and continues gradually throughout the first year of life. By 6 months of age, most of the fetal hemoglobin has been replaced by adult hemoglobin, preparing the infant's blood for the oxygen-rich environment of breathing air directly. This transition represents one of blood's most elegant adaptations, automatically shifting from one form optimized for intrauterine life to another designed for independent existence. During infancy and childhood, blood demonstrates remarkable activity as it supports rapid growth and development. The bone marrow we explored earlier works at extraordinary capacity during these years, producing new blood cells at rates that exceed those of adult life. Nearly every bone in a child's body participates in blood cell production, creating the abundant cellular supply needed for rapid tissue growth and the development of immune competence. The blood volume of children increases dramatically as they grow, but interestingly, many blood values remain remarkably stable when adjusted for body size. Hemoglobin levels, white blood cell counts, and platelet numbers maintain fairly consistent relationships to body weight throughout childhood, demonstrating how blood production scales beautifully with growth requirements. Childhood represents a time of immune system education, and blood plays a central role in this process. The white blood cells we met earlier, those protective guardians, encounter countless new substances during childhood as children explore their environment. Each encounter with harmless environmental substances helps train the immune system to distinguish between truly dangerous invaders and benign foreign materials, gradually building the sophisticated recognition capabilities that will protect health throughout life. The lymphocytes in children's blood show particularly high activity during this period of immune education. B cells produce antibodies against new antigens encountered through normal childhood experiences, while T cells learn to recognize the molecular patterns that distinguish healthy self from dangerous foreign substances. This immune education mediated through blood-borne cells creates the immunological memory that prevents most people from getting the same infectious diseases multiple times. Adolescence brings dramatic changes to blood composition and function, driven primarily by hormonal changes that prepare the body for reproductive maturity. The onset of puberty triggers increased production of growth hormones, sex hormones, and other chemical messengers that circulate through blood to coordinate the physical and physiological changes of this developmental period. For adolescent females, the beginning of menstruation introduces regular blood loss that requires adaptive changes in iron metabolism and red blood cell production. The body learns to increase iron absorption from dietary sources and enhance red blood cell production to compensate for monthly losses, demonstrating blood's remarkable ability to adapt to new physiological demands. During these teenage years, blood volume increases significantly as body size reaches adult proportions. The cardiovascular system adapts accordingly, with heart size and pumping capacity increasing to maintain optimal circulation through the larger body mass. These changes happen gradually and automatically, ensuring that the transport functions we've explored continue efficiently, despite rapid physical growth. Young adulthood represents the period when blood composition and function reach their optimal efficiency. The hematopoietic system we explored earlier operates at peak capacity, producing blood cells with maximum efficiency and maintaining ideal balances between different cell types. Red blood cell production matches destruction perfectly, white blood cell responses are rapid and effective, and platelet function ensures optimal hemostasis when needed. The blood-forming capacity gradually becomes more selective during early adulthood, concentrating primarily in the larger bones as yellow marrow replaces red marrow in many smaller bones. This transition doesn't reduce blood-forming capacity, but rather concentrates it in locations where it can operate most efficiently throughout adult life. Pregnancy represents one of blood's most remarkable adaptations, demonstrating its extraordinary capacity to meet dramatically increased physiological demands. During pregnancy, blood volume increases by approximately 40 to 50%, requiring massive increases in plasma production and red blood cell manufacturing. The heart adapts by increasing its output, while the bone marrow enhances production of all blood cell types to meet the needs of both mother and developing baby. The composition of pregnant blood also changes to support fetal development and prepare for delivery. Clotting factors increase to reduce bleeding risk during delivery, while certain nutrients like iron and folate are prioritized for transport to the developing fetus. These changes happen automatically, guided by hormonal signals that coordinate the mother's physiology with the needs of pregnancy. As we move into middle age and beyond, blood systems demonstrate continued adaptability while showing subtle signs of the aging process. Red blood cell production remains adequate, but may become slightly less efficient, while white blood cell responses may become somewhat less rapid and intense. These changes happen so gradually that they rarely interfere with normal daily activities, representing natural adaptations to the aging process rather than pathological changes. The bone marrow's blood-producing capacity shows remarkable resilience throughout life, maintaining adequate production of all blood cell types well into advanced age. While the total volume of active bone marrow may decrease somewhat, the remaining marrow compensates by increasing its productivity per unit volume. This adaptation ensures that blood cell production continues to meet physiological needs throughout the human lifespan. Older adults may experience subtle changes in blood composition that reflect the accumulated effects of decades of life experience. Iron stores may increase gradually over time, while certain vitamins may require more attention to maintain optimal levels. The immune system's memory, carried by long-lived lymphocytes in blood, may contain antibodies against dozens of different infectious agents encountered over a lifetime, creating a vast library of immunological experience. The transport functions of blood remain remarkably stable throughout life, continuing to deliver nutrients, remove wastes, and carry chemical messages with efficiency that supports active, healthy living well into advanced age. The physical properties we explored, viscosity, temperature regulation, and circulation, adapt gradually to age-related changes in cardiovascular function while maintaining the essential characteristics that make blood an effective transport medium. Understanding how blood changes throughout life helps us appreciate both its remarkable adaptability and its fundamental stability. The same basic processes that sustain a newborn also sustain an elderly person, adapted and refined through decades of faithful service. The red blood cells carrying oxygen, the white blood cells protecting against infection, the platelets preventing bleeding, and the plasma carrying nutrients all continue their essential work throughout the entire human lifespan. As we prepare to explore the future possibilities that may enhance blood's natural capabilities, take a moment to appreciate this lifelong adaptation that's occurring within you right now. The blood flowing through your vessels has already adapted to your current stage of life, meeting your body's needs with the wisdom accumulated through millions of years of evolutionary refinement and your own lifetime of experience. Allow yourself to feel grateful for blood's remarkable capacity to grow with you, adapt to your changing needs, and continue its vital work regardless of your age or stage of life. This adaptability ensures that the complex transport, protective, and regulatory functions we've explored together will continue to serve you throughout your entire lifetime, adjusted and refined to meet whatever challenges and opportunities life may bring. Let this appreciation for blood's lifelong adaptability settle peacefully in your mind as we prepare to explore the exciting possibilities that future medical advances may bring to support and enhance these remarkable natural systems. As our peaceful journey through the remarkable world of blood draws toward its gentle conclusion, let yourself settle into this final state of calm learning. We now turn our attention toward the future, not with excitement or urgency, but with quiet hope and wonder at the remarkable advances that may soon enhance blood's natural healing capabilities. In this closing exploration, we'll softly discover how scientists and medical researchers are working to understand blood even more deeply, developing therapies that partner with the extraordinary systems we've been exploring together. Think of these future possibilities, not as dramatic departures from nature's design, but as humanity's growing partnership with blood's inherent wisdom. Every advance in blood science builds upon the fundamental processes we've discovered: the gentle oxygen transport of red blood cells, the protective vigilance of white blood cells, the healing dedication of platelets, and the sophisticated chemistry of plasma. Future therapies seek to support, enhance, and sometimes restore these natural capabilities when they need assistance. One of the most promising areas of advancement lies in regenerative medicine, particularly the use of stem cells to enhance blood's natural renewal capabilities. Remember our exploration of the bone marrow, that hidden factory where all blood cells begin their lives from pluripotent stem cells. Scientists are learning to harness these remarkable cells' potential to treat conditions that were once considered incurable, offering hope for people whose blood-forming systems have been damaged by disease, chemotherapy, or genetic conditions. Stem cell therapies work by providing fresh, healthy stem cells that can re-establish normal blood cell production when a person's own bone marrow has been compromised. These treatments don't fight against natural processes, but rather restore them, allowing the body's own healing wisdom to resume its work. Current research is exploring how to make these therapies more effective, more widely available, and applicable to an even broader range of conditions. The field of gene therapy represents another gentle but profound frontier in blood medicine. Scientists are developing ways to correct genetic variations at their source, potentially offering permanent solutions for inherited blood disorders. These approaches work by providing cells with correct genetic instructions, allowing them to produce normal proteins and function as nature intended. For conditions like sickle cell disease, where a single genetic change affects hemoglobin structure and red blood cell function, gene therapy offers the possibility of giving stem cells the correct instructions for producing normal hemoglobin. Early clinical trials have shown remarkable success, with some patients achieving complete freedom from the complications of their condition after receiving corrected stem cells. These treatments represent hope for thousands of people worldwide who live with genetic blood disorders. Artificial blood substitutes represent another fascinating area of research, as scientists work to recreate blood's remarkable transport properties in laboratory settings. While no artificial substitute can fully replicate the complexity of natural blood, researchers are developing products that can temporarily support oxygen transport during emergencies or surgical procedures when donated blood isn't available. These artificial carriers often use modified hemoglobin molecules or synthetic compounds that can bind and release oxygen similarly to natural red blood cells. While still in development, they offer the possibility of universal oxygen carriers that could be stored long-term and used immediately in emergency situations, potentially saving lives in remote locations or during disasters when traditional blood banking isn't possible. The science of precision medicine is revolutionizing how blood disorders are treated, moving away from one-size-fits-all approaches toward therapies tailored to individual genetic profiles and specific cellular characteristics. By analyzing the precise molecular details of a person's blood disorder, physicians can select treatments that address the specific mechanisms causing problems in that individual, often achieving better results with fewer side effects. This personalized approach extends to cancer treatment, where understanding the specific genetic changes in a person's leukemia or lymphoma cells allows physicians to select targeted therapies that attack those particular cellular abnormalities while preserving normal blood cells. Many of these targeted treatments work by blocking specific cellular signals that cancer cells need to survive and multiply, allowing normal blood cell production to resume. Immunotherapy represents another promising frontier, harnessing the power of the immune system we explored earlier to treat various conditions. Scientists are learning to train immune cells to recognize and attack specific targets, from cancer cells to infectious agents that resist traditional treatments. Some approaches involve removing immune cells from a patient's blood, training them in the laboratory to better recognize threats, then returning them to circulation where they can work more effectively. CAR T-cell therapy, where T cells are genetically modified to better recognize and attack specific cancer cells, has achieved remarkable success in treating certain blood cancers that didn't respond to other treatments. These approaches work by enhancing the natural protective capabilities of white blood cells rather than replacing them, representing a partnership between medical science and natural immune function. Advances in blood banking and transfusion medicine continue to improve the safety and effectiveness of these life-saving procedures. Researchers are developing better methods for storing blood components, extending their useful life while maintaining their function. New testing methods can detect infectious agents more quickly and accurately, making transfusions even safer than they are today. Artificial preservation techniques may soon allow blood components to be stored for much longer periods, potentially solving supply shortages and making blood products available in areas where traditional banking isn't feasible. Some research focuses on freeze-drying blood components or using other preservation methods that could create stable, long-term storage options. The future of blood testing promises even more detailed insights into health and disease through advances in analytical technology. Liquid biopsies, which detect genetic material from various organs circulating in blood, may soon allow early detection of cancers and other conditions before they become symptomatic. These tests work by identifying molecular signatures that indicate disease processes beginning in specific organs, turning blood into an even more powerful diagnostic tool. Advances in nanotechnology may enable microscopic devices that can circulate in blood, monitoring various parameters continuously and providing real-time information about health status. While still in early research phases, these technologies could transform how we monitor and manage various conditions, providing instant feedback about treatment effectiveness and early warning of potential problems. Research into blood's role in aging and longevity explores how changes in blood composition over time might be addressed to support healthy aging. Some studies examine whether young blood factors might help restore function in older organisms, while others investigate how to maintain optimal blood composition throughout the lifespan. These investigations don't seek to stop aging, but rather to understand how blood's natural functions might be supported to maintain health and vitality longer. The field of tissue engineering is exploring how to create blood vessels and even entire organs using a person's own cells, potentially solving the shortage of organs available for transplantation. These approaches often use stem cells from blood or bone marrow as starting materials, growing them on artificial scaffolds that gradually dissolve as natural tissues form. International collaboration in blood research ensures that advances benefit people worldwide, with researchers sharing knowledge and resources to accelerate progress. Global initiatives work to make new treatments accessible regardless of geographic location or economic circumstances, recognizing that advances in blood medicine represent improvements in fundamental human health that should benefit everyone. As our gentle journey through the world of blood reaches its peaceful conclusion, allow yourself to feel hopeful about these continuing advances in understanding and supporting the remarkable systems we've explored together. Every breakthrough builds upon the foundation of knowledge we've shared, deepening appreciation for blood's natural capabilities while developing new ways to support them when needed. The future of blood science represents not a replacement of natural processes, but rather an enhancement of them, supporting the oxygen transport we've discovered, strengthening the immune protection we've witnessed, improving the healing capabilities we've appreciated, and maintaining the chemical balance we've understood. These advances offer hope for people whose blood systems need support, while deepening our understanding of the remarkable fluid that sustains all complex life. Allow yourself to feel grateful for the researchers and medical professionals who dedicate their lives to understanding and supporting these vital systems. Their work ensures that future generations will benefit from even better treatments, more effective preventions, and deeper understanding of the remarkable fluid flowing through every one of us. Let this gentle hope for the future settle peacefully in your mind, concluding our journey with gratitude for both the natural wonder of blood and humanity's dedication to supporting and protecting this precious gift of life.